Okay, so going back to where I stopped. So when we measure the coevolutionary trajectories of those attack and defense traits and considering a local network again, so we found that the structure of the network actually had a strong effect on the coevolutionary trajectories. So in the case of networks that are highly connected and that form this pattern of interaction, which we call a nested pattern, because we have generalist species interacting among themselves, and the specialist species, they don't form small groups of species, they interact with this core of generalist species. So in this case, we would expect to find a much lower attacking defense traits arms races than when we have networks that are less connected and that tend to form these small groups of interacting species. So this kind of structure that you see here in the left would be favoring escalating arms races in parasite host networks. On the other side, if we look at another measure of the coevolutionary trajectory, which is the amount of fluctuating selection, we found that highly connected networks that have this kind of nested pattern here in the right, we would find much more fluctuating selection dynamics than when we have more less connected networks, and those networks are more modular because they tend to form these small groups of interacting species. And this means in a multi-species context, fluctuating selection is what we also call coevolutionary alternation, because what happens is that a parasite is changing the host species that it is affecting at each time according to relative levels of attack and defense traits. So it is attacking much more the host species that has lower levels of defenses. And then in the next step, when you have a coevolutionary response to that, then it is kind of shifting the host species that is attacking at each time step. And when we look at not only the variation in traits but also abundances, what we found is that when environmental selection is stronger than interaction selection is coevolution, then we have species abundances fluctuating in time in response to these different environmental selection pressures. But species traits tend to be much more stable than population dynamics. But on the other side, if interaction selection is stronger, that is, if coevolution is the main driver in this system, then we will find much more fluctuating selection dynamics there, and these kind of stabilizes population dynamics, and this is increasing the stability of those systems and can be also a mechanism by which we keep in networks a high diversity of parasites there, because fluctuating selection is favoring the demographic stability of these and favoring also higher diversity because the species are less prone to local extinctions. But this is at a local network. If we think about a meta-community context in which forest patches and networks are linked by the dispersal of some species, we still don't know what are the coevolutionary outcomes in this context, and this is one of the main questions in this project. What we do know, and this is coming from the geographic mosaic of coevolution theory by John Thompson, is that when we have when species interact with different communities in each patch, then we will expect also different coevolutionary reciprocal selection pressures in each one of these patches. So it could be that in some patch we have only selection in one species, like only selection in the hosts. In other patches we can have selection acting only in the parasites, but we can also have patches where reciprocal selection pressure is very strong in both species, and this is in these places would be happening the coevolutionary hotspots, but also reciprocal selection pressure could be weak in both species, and there we will find coevolutionary cold spots. And this mosaic of different selection differentials in the landscape would lead to a much more complex coevolutionary trajectory in these parasite host systems. And we also don't know what is going to happen in this case. This is one of the main questions also here in this workshop. We want to develop these. But it has already been suggested that this coevolutionary effect would be a main driver of spillover, because having these different trajectories in different patches that are linked would increase the probability that a zoonotic pathogen would spill over to different host populations. But this is an opinion paper, and this is an hypothesis. So this is a question, and this is what we would like to be doing during this week here in the workshop. What we do have is that we have a lot of data coming from different land uses. We have data on species interactions and data on traits and phylogeny, so that we can adapt these adaptive network models to produce spillover probabilities across the landscape. This I'm bringing Brazilian data, but obviously we can apply these to many other places and contexts. So thank you very much. I don't know if we have time for questions. Maybe we should go to the next talk and now let's see if we have questions. Okay, so we have maybe 2-3 minutes for questions.
Oh, I think it's working.
Thank you. Nice talk. I'm not sure about the time scales that we're talking about here. Are they very separated, the ecological time scale and the evolutionary time scale? Are they separated, or they are
No,
comparable?
we're talking about what we call current evolution. So they occur at the same time scale because, well, in the case of these interactions, actually the coevolution affects the epidemiological trajectory because you can have novel strains. So I'm not talking about speciation and host attack. So I'm talking about trait evolution, which.
Yeah. Okay. So the changes in the composition of the populations are the kind of evolutionary changes you're talking about, right?
Yes.
Okay. Thank you.
Great talk, very nice problem. I have also the same question. And also another one is related with the bipartite network that you show in each patch. From the picture, it seems that the structure is the same in all of them. The only thing that is different is that some species or some traits are present or not. Can vary? I mean, it's a matter of the picture, or the structure of this network can vary across patches because of the I don't know, environmental thing or whatever.
Thank you for this question, because actually this is Mariana's talk. Yes, actually the structure change. It's not necessarily that we're going to find the same pattern in each one of these patches, and it's greatly affected by the size of the fragment, the quality, and well, Mariana is going to tell us a little bit about this later.
We are already late, so one more. Very nice talk, Cecilia. Sorry. So you talked about amplification and dilution effect, and you also talked about the theory from John Thompson. Could we relate the amplification and the dilution effect with hotspots and cold spots of coevolution? Do you think this amplification effect, for instance, is related with a hotspot for the coevolutionary process?
We don't know that, but in this perspective paper, which is I really like this paper because it was like bringing these hypotheses that maybe in those places where you have these amplification hotspots, this amplification effect, they would be coevolutionary hotspots and like sources of new variants to other places. This is their hypothesis.
Their hypothesis. Yeah. Okay, so I understand better now. Okay, thank you.
So, thank you for your opening remarks, Cecilia, for the following talk. So, I would like to introduce you to Dr. Javier Pérez-Tris. Javier is a professor and co-director of the Evolution and Conservation Biology Research Group here at the Complutense University of Madrid, as well as the university's representative of the Self-Steering Committee of Una Europa, One Health. His research integrates concepts and methods of evolutionary biology, biogeography, behavioral ecology, and conservation biology to investigate the causes and consequences of the phenotypic diversity of animals. Please.
Thank you very much. Let's see if I find my presentation somewhere. Here it is. Great. Thank you very much for the presentation, and thank you Cecilia and Giselle and Ana Paula for inviting me to this meeting. It's really great to have this at the Complutense University. So this is really a game changer, I think. So thanks for that. We will talk about that later. But my presence here is to talk about a case study. This is the bird-malaria interactions. It's not really like the best case for disease transmission if you think of zoonotic diseases that affect humans and stuff, but it can be a good example of a specific study system in which to apply the concepts and the tools that we will talk about in this workshop. So just to present the is this working or not?
No, it's not.
Okay, so we have to doesn't matter. Just to present the parasites. So what are these avian malaria parasites? Well, actually, in the broad sense, is what we call bird emersporidians. They are formed by different genera of parasites. Plasmodium is the most virulent, according to most of the literature. Is very similar to the parasite that affects humans, but it's not the same species that are affecting birds. And then other genera, Hemoproteus and Leucocytoson, they have also high prevalence and diversity in birds. Collectively, we call those avian malaria parasites, and they work more or less the same, although they have a lot of distinct characteristics that make them unique. So what's the basics of this interaction? So malaria parasites are vector-borne protozoan parasites, so they are transmitted by vectors into the birds. The birds are the intermediate hosts, so there's a cycle within the bird, and then the last step in this cycle is the formation of gametocytes, which are the transmissible phases of the parasite. So the mosquitoes biting infected hosts that are competent hosts, they take the gametocytes and then they complete the cycle. So it is the idea is that we need birds that are competent, we need the parasites, and we need vectors that are also competent in transmitting the parasites, and this changes across different scales. This is just to show you what. Yeah, I stay here. No, don't worry. Thanks. It's just to show you the evolution of publications on avian malaria. At the beginning, it was a model system for the understanding of human malaria, and this is why it started to gain attention. But then during many years, there were a handful of studies per year. They were based on microscopy, and then at some point, basically around the starting of the 21st century, the incorporation of molecular tools increased the attention on these parasites, because it made it very easy to study the interaction even for non-specialists. So many people started to do PCRs on bird blood samples and to identify different parasites in the blood. And then some decade later of this change, this database, this resource, Malawi, was released. This is a repository, a sort of a gene bank repository of malaria parasites that includes information on the distribution across hosts, across populations, and I mean as detailed as possible. It's increasing in detail every day basically, and it's a good resource for studies of avian malaria. So the basics of this tool is that the parasite identity is determined by cytochrome B DNA barcodes. So we screen the blood of the birds and find fragments of the parasite DNA that we can sequence and identify which species of the parasite is in the blood. There's an advantage of this method, that is that all parasites are in principle searched for in each individual, so every link that is possible is detected, or well, it depends on, at least in theory, it depends on many things, but most of the times the parasites that are there are found, and so far there are over 4,000 unique sequences that have been described. This we call parasite lineages. And then what are those lineages? Because at the beginning one could think that they could be intraspecific polymorphisms, but if we look at the relationships between mitochondrial, the marker we use for parasite screening, and nuclear genes, we see that there's a correspondence that is compatible with or supports very well the idea that these are biologically distinct entities that can be treated as a species. And actually it's not I mean, this is one of the first studies that show that, and other studies have shown that they have phenotypic diversity, that they have different transmission areas or transmission periods, they have different specificity, and so on. So these parasites that we identify by the sequence are biologically distinct, and then we can look at the network of interactions with different hosts in different areas. So if we look at in general to the distribution of the parasites, what we see is that most of the parasites are specific. So most of the parasites have been found in just one bird species, 72% so far. But a few parasites are highly generalist. So some of them have been found in hundreds of species, and they are found anywhere in the world. They are invasive. They have a very different biological interaction with their hosts. So one of the first ideas was to look at how this was structured. And thinking of the specific parasites, this specificity can be maintained in the long run evolutionarily. So, for example, this group of parasites here, these are called Hemoproteus parabellopolski, affect different birds that are closely related. This is the three birds that are typically affected by this group of parasites. Is the black caps, the garden warblers, and the Abyssinian warbler. And the parasites, for some reason, have evolved within this group. So they have changed host in evolutionary time sometimes, but they maintain in ecological time restricted to one host each. So each species of bird has its own parasites, and there's very little interchange. Only one of those parasites has been found in two of the three species. So this tells us that the parasites are able to change host every now and then, but they maintain their interaction with one host in the long run in this system. Then this creates also some evolutionary stability in transmission dynamics, for example in transmission areas. So if you look at the parasites that are found in the European birds or in African birds, and in birds that migrate between Africa and Europe, then you see that some parasites can be said to be transmitted only in Europe if they are found only in birds in Europe but never in African birds, or they are transmitted in Africa when they are found in African birds or in migrants that go to Europe, but in those cases the parasites are found only in the adults of those migrants. So in the juveniles that never visited Africa, you don't find those parasites. So you can look at the prevalence of different parasites in different sorts of sets of birds, and then the pattern that emerges is that parasites are either transmitted in Africa or in Europe. It's in red or blue in this graph, and only a few parasites, like the one in green with the arrow, are found to be transmitted in both areas. So this means that even if we are looking at the blood of the migrants that come to Europe every year, and they carry the parasites acquired in Africa in their blood, those parasites for some reason don't make it out to the local European fauna, so they don't spread very often. So they're like knocking on the door but not entering the system. So these results would tell us that the parasites are restricted to hosts and to transmission area, and they are they have trouble to really spill over across species. But then if you look at the evolutionary relationships between the parasites and the host in a broad scale, what you see is that you have to assume a lot of host switches across the evolution of this interaction. So the idea is that they may change every now and then. And actually, at the same time, we see that there's a lot of host switching in ecological time, because if you look at local communities, this is a result of a distribution of different parasites in the local communities of reedbed pasturines in Portugal. You see that the same parasites are found in different species, and genetically they are the same parasites actually. So if you look at more detail into the genes, they seem to be the same changing between hosts. And most importantly, from this perspective, is that exotic birds that have been imported into the system acquire parasites from the system. So this means that the parasites are ready to jump into new species when they have the opportunity, and this is very well documented by these observations of the exotics having the parasites of the local birds. So if that means that thinking, for example, of problems like invasions and colonizations may tell us about the opportunity of a spillover of those parasites. So we looked, for example, at this system, which is the birds that have been introduced in New Zealand and the local birds from New Zealand, and we looked at the parasites that those birds had, and what we found is that we only found 8 Plasmodium parasites in this system, and they were distantly related, so it was nothing like a monophyletic group of New Zealand parasites, but they were picked from different parts of the phylogeny, and they had like a different, at first glance, a different specificity. So the length of the bars here in this graph, each tip is a parasite. The length of the bar is a measure of the host range of the parasite. So the bigger the bar, the more hosts a parasite is able to colonize. And then the colors, look, they represent countries or areas of the world where each parasite has been found. So just here is easier to see if we look at the degree of specificity of these parasites that have been found colonizing the native avifauna of New Zealand, or we look at the distribution across the world of those parasites, or we look at the prevalence in the native areas in Europe where the birds that were carried to New Zealand came from, we see that the community that got established in New Zealand is much more generalist, much more broadly distributed, and much more prevalent in native areas than you would expect by a random sampling of the diversity of parasite candidates to colonize. So this tells us that there are some traits of the parasites that make them more prone to colonize new birds. So the generalists are probably the ones that are more prepared to spill over other species. Even if in the local system it seems that they are still dependent on the exotic birds to maintain their transmission dynamics. So the main reservoirs of these parasites, of these exotic parasites in the exotic area, are the exotic birds. So this is something we still don't know very well how it works, but it could be an opportunity to look at how the spillover probability or the spillover capacity of the parasites would depend also on the characteristics of the network of interactions with other hosts. So they need probably the support of their native hosts to get established during the period of establishment. So this is something that could be possible. If we look at what happens on islands, for example, if we come back to the black cap, which is a parasite that has a lot of parasites that are actually specific of that species, if we look at the populations on oceanic islands that were colonized recently, like 30,000 years ago or so, then we see that there's a lot of parasites that disappear from the system. So there's most of the parasites just disappear, and what are lost are the specialist parasites. Only the generalists persist in the populations at very low prevalence. And the interesting part of this study is that in this system, in which a lot of parasites have disappeared from the colonizing host, new parasites have been acquired locally. So and those parasites are also generalists. So it seems that the degree of specialization limits the capacity of parasites to colonize new hosts. And then I will talk a bit about the traits of the parasites and the hosts, because one problem with this system is that sometimes with the tools we have at hand, it is difficult to know exactly how generalist or how specialist a parasite is. And this is a very illustrative case of that. So if we look at the distribution of this Haemoproteus witti, which is the most generalist of all Haemoproteus parasites, it is found in lots of different bird species. It's a neotropical parasite, so through the forests of the Andean mountains in South America, you have lots of different species from different families and orders infected by this parasite, so you can detect the sequence in the blood. But then, when you look at the at the intensity of the infection in each different host species, in this graph, each dot is one species, and all the black dots, which are basically all the dots that you see, are hummingbirds, so which is the typical host of this parasite. They have a variation in intensity, so some of them have a low intensity, some of them have very high intensity, up to 25% of the erythrocytes can be infected with gametocytes, and it's very easy to see the gametocytes in the blood of these birds. But then all the other birds of the other species or of the other families are all together in this gray point down there in the red cycle. So this is not just one point, but a lot of different species that have very low prevalence. They can be detected in the blood, but they have very low prevalence. And when you look at the blood of those birds, you don't see any gametocytes. So they seem to be infected. They probably suffer from the infection. We don't know how virulent this infection can be to them, but they are not competent hosts in the sense that they can transmit the infection to other hosts. So there's a lot of spillover that may have health consequences for the hosts, but not for the transmission of the parasite at the local level. It may have consequences in terms of dilution effects and stuff like that, but we don't know exactly how it works. And this may be still more clear, for example, in other cases, and this we expect this to happen. I mean, this is just one example in occasions in which parasites are put into contact with new hosts or new parasites with new hosts. So this is a problem that has happened recently in Europe with exotic parakeets and parrots that are infected by a blackbird parasite, basically. So it's a parasite that is very common in thrushes and blackbirds here in Europe. But when it infects this parrot, it goes into the organs and tries to complete the cycle, but for some reason it cannot complete the cycle. So it seems that something in the physiology of these birds is blocking the parasite from developing into blood stages, and then you never see those parasites in the blood. You never detect them by PCR or any method. So it's like they seem not infected, but then when you look at the organs when they die, they are packed with microneurons and, well, like these structures that kill the bird. And the idea is that for some reason that we don't understand yet, so it's probably related with the physiology of the host and the traits that affect the infectivity of the parasites, they are forbidden links. So you have some interactions that cannot be established, even if there's an opportunity to establish those interactions. So thinking of all those things, recently we have been looking at the distribution of parasites in a mega diverse community in French Guiana. So the idea is to look at one single place where all the parasites are locally transmitted. So the good thing with this place is that no migrants occur in the area, so all the birds are local, they don't move, and they need to have acquired the parasites locally. So this is the transmission that happens there is between the birds that are there. Everything happens in French Guiana, it stays in French Guiana, and that's it. So the thing is that if you look at the distribution of the parasites across hosts, there's a clear pattern in which some species are accumulating infections and other species for some reason are escaping infections. So in this graph, the gray dots represent the expected prevalence of the parasites in each species given the abundance of the species. So the more common the bird, the easier should be that a parasite hits that species. And if you distribute the observed infections across the species randomly, then you would expect a distribution of the parasites, like raining over the hosts. But then you see that some birds that are marked with this red stat accumulate many more infections that they would acquire by chance. And perhaps more interesting, or well, with the same interest, some birds that are very common, they never get infected, or well, very seldom they get infected. So this distinguishes between avoiders and key hosts, and that could be important in terms of how the interaction is maintained and how it can evolve if different species, for example, disappear or enter these systems. So at that point, it is important to look at what makes a good host. For example, so we look at the traits of different species that there are hosts of these parasites, and just code different traits that we could get from the literature, like body size, diet, morphological traits that expose those birds to vector bites or habitat choice and stuff like that. And we found that there are patterns. So this is very preliminary because we have few species and we don't have that much power with this data set, but we are increasing the number of birds that are screened, and we see, for example, that the birds that are more exposed to vector bites or that have more, their diets are more based on plant matter, they get more chances to have Plasmodium infections in this system. So this was just to present the system, and maybe we were talking a bit with Cecilia and thinking of possibilities of developing these ideas, and we have some open questions that are very clear. So, for example, what makes a generalist parasite? So we want to look at the things that happen at the genomic level, for example, in parasites that are generalists compared to the closest relatives that are very specialist. So this is something that is now ongoing. What is a generalist host? Also, this is an interesting question. So we know that there are hosts that have very high competence to host some parasites, but we actually haven't looked yet to which hosts are really generalists. So they host many different kinds of parasites. For example, the robin here in European forests has a low prevalence in general and has a low prevalence of per parasite. So for one parasite, the robin is not a very good host, but it has many different parasites that are present in the local community and probably depend on other hosts to maintain the populations. So we also want to look at the differences in the structure of the network across different local communities. So we want to know if the different hosts and the different parasites have played the same role in different communities or in different geographic regions, because many of them are distributed across the world. And also want to know if parasite infections, this is like a jump into the whole thing, may determine the individual host roles in other ecological networks. Like, for example, if infected birds tend to feed on things that help them manage the infection, this will change the patterns, for example, of fruit dispersal or things like that. So this is basically what I wanted to tell you about this system, and many thanks for your attention, and question, please. Yes.
Thank you for the talk. Nice talk. I was wondering about your question, not make my parasite generalist or not, and is there any? Did you look for if there is any relation between old lineages and new lineage, as if there is a parasite that accumulate capability to infect, something like that.
Yeah.
Another, so can I have two questions? One is there is some relation, lineage relation, and then another one, if those parasites compete for the research, so it's more likely when you take one individual bird, they usually find just one, and the generalization would be among individuals and not in one individual.
Mm-hmm. Yeah, the first question. Apparently, what we see is that there's no. I mean, the generalist strategy seems to be very labile evolutionarily. So you have in the phylogeny of the parasites, you have some parasites that get generalist, and they don't leave many descendants. So it seems that they're always like a single tips across the phylogeny. And then actually, this idea of how generalist the parasite can be extended to the other traits. Like, for example, how generalist they are, for example, in terms of transmission periods or stuff like that. For example, in this system with black caps, we know that some parasites are transmitted in summer only, so it's like the typical period of transmission of parasites when you have lots of vectors and stuff like that. But some parasites, for some reason, they make it to be transmitted across the year, so they are present in the blood all year long, and then suddenly they have the opportunity to jump from population to population, because the black caps that live in different breeding areas, they meet together in the same wintering grounds. So the parasite that changed the strategy of transmission to be able to ride that wave makes it, it gets super successful. So they get much higher prevalence, they get a broader distribution and stuff, but they don't stay evolutionarily for some reason. So they always, this evolves every now and then. Those parasites get very successful locally and temporarily, but then they vanish for some reason. So it seems that on the long run, the specialist strategy is the most stable. So it's the one that is selected, but we don't know what happens there. But generalists are popping up every now and then. And then the other question was about competition between the different parasites. So we actually don't know what happens there. It may have there may be a lot of variation in how the coexistence of different parasites in the block, for example, when they compete for vectors or for hosts, is affecting the different parasites. We actually have some observations of parasites that coexist, and it seems that sometimes this has been shown, for example, in other systems like in rodent malaria and stuff. When the parasites coexist, they sort of they interact sometimes positively, So for some parasites, it seems that they need the host to be infected by other parasite to really be able to colonize. But sometimes it's a negative interaction. So when they coexist in blood, only one produces viable gametocytes that are taken by the mosquitoes. The other one is present there. You can see the parasites. You can see the different parts of the cycle stuff. But only one is making it out of the host. And this is something that is still not really known. But yeah, that's fascinating, really. Mm-hmm. Oh, yeah.
Yeah, I have so many questions. So when you were talking about this competition, I think inside the host, when you see that only one of these strains, I don't know, for instance, only one parasite stays, is it the generalist or the specialist? Do you know the answer?
It may be any of those. So it can be one or the other one. So you find them, for example, with the black cap is kind of easy to study that because you have most of the birds are infected, so sometimes 100% of the population has infections, and many birds have many different infections in the blood. So you may have from one parasite to up to 5 different parasites in the blood mixing there. And we know that there are different species, so each parasite has its own point of view about how much the bird has to be exploded and exploded and all this stuff. So sometimes you have only a specialist, sometimes you have only generalists, sometimes you have both together. And what I don't know if it's we don't have good data to analyze that. So at the moment we may have at some point if we analyze some samples that we have. but we don't know how they change in the blood of. So one thing we want to know is we have been putting a lot of effort in the last years, sampling the same population in a longitudinal project, like having the same individual bird sampled through the year many times and across years various times to see how the infection is changing. So if one parasite is increasing and the other one is decreasing, or what are the intra-host dynamics of the parasites, to see if some parasites are better fighters from that perspective or not. We don't know. I could expect that this may happen because, as I said before, for example, in rodent malaria, there's one parasite is called Plasmodium chabaudi that is able to block the transmission of other parasites when it occurs in the blood with this other parasite. But I don't know if this happens in birds. There was if I can elaborate a bit. There was a very nice research that was done by Gerd Balkunas, who is the master of these parasites. He was doing what he calls exflagellation experiments. So you take the blood sample of the birds and put it on air, and then the parasites, the gametocytes, develop into gametes, and they fuse and make the okinete that will colonize the bird. But you can see if they are viable or not. And he wanted to do this compatibility studies, like between different parasites and stuff. And what he found is that when you have single infections of parasites of 2 different lineages, they abort a lot of gametocytes. So they not the gametocytes of gametes. So they fuse, and then many of the gametes they just suicide or something. So they get aborted when they are of the same lineage. But then when you mix lineages, what it seems is that the level of abortion is much lower. So it's and this was counterintuitive because he suspected that the mix of lineages would cause like a barrier against hybridization and more abortion. And then he found exactly the opposite, and that was compatible with the idea that when they are mixing together, the pressure to really make it to the vector is much higher, and then they put all the effort into making gametes, while when they are alone they have the vector from the sun, so they can actually hit with a lower impact on the vector by aborting parasites. So it seems that there's a lot of between parasite dynamics going on in there, but we don't understand them well for the moment.
Okay, thank you. I have many questions, but I think
So
I can make during coffee break.
And we can talk later. Yeah. So
Yeah.
whatever you want, but
Thank you.
yeah, thank you very much.
Anybody else? Just one?
Hi, thanks for a great talk. So my question is more about the virulence. So what do you think makes both a good generalist parasite and a generalist host regarding, or with regards to the virulence? Because, you know, for example, with the invasive species, like there's a risk and a trade-off between what's been known that has been happening with the Hawaiian bee birds and but in terms of evolutionary arms race, I guess being very virulent is not good for a parasite that tries to be consistent in time.
Yeah, I don't know. I don't know if there's a correlation between virulence and degree of specialization. There's a correlation, for example, with prevalence. That's for sure, and that makes sense because of the dynamics of the transmission. But the virulence, I think, it depends on the very interaction between the parasite and the host. So the same parasite in different hosts has a different virulence. So it's an interaction trait and not a parasite trait. Even if I think, this is my guess, that some parasites are more prone to be virulent than others. So this is, for example, one example is the example with the Hemoptyus minutus with the parrots. I mean, it's super virulent. It's lethal to the parrots, but you see every blackbird around here is carrying that parasite. So it's like it's not a parasite trait, but and thinking, for example, of invasions and these systems in Hawaii, for example, the parasite that entered Hawaii for some reason was very, very virulent. Probably it was because this is well studied. The host didn't have the means to resist the parasite, and there was a lot of selection against susceptibles and the discipline of the host changed and stuff. But now they are starting to get adapted to the parasite and recolonizing the areas where the mosquito is living. So given some time, it may be adapted. So I don't think it's. I actually don't know what's the problem for the generalist parasites to persist. But I think it has more to do with the transmission dynamics than with resistance or virulence. Yeah. That's my guess. Thank you. We continue.
Thank you, Javi, for the great talk. I invite you all to the coffee break now, and we will return in roughly 20 minutes. Thank you. Sorry, everyone. Another technical issue, so we'll have just 10 minutes of coffee break, okay? Thank you. So welcome everyone. Welcome back. For the following talk of our event, we have Dr. José Cuesta. José is a professor in the mathematics department of the University Carlos III of Madrid and leads the complex system interdisciplinary group at the same institution. Please, Dr. José.
Thank you. Thank you. Thank you for the invitation and for the presentation. I'm sorry, I work in the mathematics department, so but my research is related to biology. I'm a theoretician, of course, and well, this title more or less gather basically the 2 lines of research I'm conducting in biology right now. Before we start, let me just briefly introduce my group. The first row is people related to ecology. The second row is people related to evolution. Jose and Nelo are my two closest collaborators in ecology, and Mateo and Miguel are recent collaborators in the University of Granada. Then in evolution, I work for many years with Susana Manrubia and her group, to which Toño belongs. And then there's also Pablo and Pilar, who are in Carlos III, and Jacobo Aguirre, who is in the Center of Astrobiology. Okay. Basically, I'm going to introduce 3 research lines very briefly, and I'm going to talk about 3 representative problems I'm working on in these topics. The first is microbial ecology, the second is genotype-phenotype maps, and the third one is gene-genome networks. All of them are related to networks to some extent, and the dynamics on these networks, which is the key interest of my research. Well, first, microbial evolution. If I have to explain why I am interested in microbial evolution, I have to say the standard thing that the world is full of bacteria. Our body, there are as many cells, human cells, as there are bacteria. Also, you can find bacteria that bacteria influences in everything, in the health, in the mental state, in many things in our body. But you can also find bacteria everywhere, in the soil, in the water, in the clouds. But if I have to be honest, the reason why I'm studying this is microecology. Microecology is a recent comer to ecology, and the idea of microecology is that it studies large-scale patterns. And bacteria, fortunately, are large-scale ecosystems, and then you can find patterns that you recognize, statistical patterns, and these statistical patterns are interesting to me, at least, because they reveal underlying dynamical processes. And this is the thing that we're interested in. We want to identify statistical patterns and then try to identify what are the mechanisms, the dynamical mechanisms producing these patterns. Just as an example, a very recent example, this is a work of Jacopo Grilli in 2020. He identified this pattern in microbes. What he did was he studied an ecosystem, let's say, for instance, the gut, and then he studied different samples in different communities in the gut, right? And for each species in these communities, he recorded the abundance. And what he did was, well, the standard, let's say, the vertical distribution would be the standard species abundance distribution, but what he did was just taking across these communities. So you take a species across communities, and then you study what is the distribution of abundances there. And this distribution is called the abundance fluctuations distribution, because these are the fluctuations between different communities of the same species, right? What he found is that they follow a common pattern that you can see down there. All of them, for every species, what you find is that this distribution follows a gamma distribution. Then there's another pattern. If you make averages across communities, then you take the average and the variance, and then you compare the mean and the variance, and what you see is that they follow a power law. And this power law is basically one of the examples of Taylor's law, right? So basically the variance behaves as the square of the abundance, or the average abundance. And also, he found that the distribution of the means follows a known pattern, which is a log normal distribution, or a truncated log normal distribution, because, well, to the left species are too rare to be observed, right? But the part that is observed follows very well a log normal distribution. So these are three typical statistical patterns that you have to explain through dynamical processes. And he introduced, to explain then, this model, which is the stochastic logistic model, which is basically a logistic model, a standard logistic model of growth, but then you introduce environmental noise through this term. It is environmental noise because it's proportional to the abundance of the species, and this is actually the kind of fluctuations that you observe. And basically, the trajectories that you observe are like jiggling trajectories of the standard sigmoid that you observe in the logistic growth, right? Okay, but there is another pattern that you can observe in these communities, which is when you compare the abundance distributions of two different species, or the abundances of two different species across communities, and then you can compute the Pearson correlation coefficient. So you observe the correlation between species, and you can observe that they are not correlated at all, or that they have positive correlation or negative correlation, depending on these coefficients. So Jacopo Grilli also studied these correlations, and he computed the distribution of Pearson coefficients in different ecosystems, and what he found was these color curves here in different ecosystems, and in black you can see what you obtain if there are no correlations. So this is why the spurious correlation that you observe in the data when you have just uncorrelated data, right? So you can see that there are actual correlations, something that has to be explained and something that you can't explain with the model he introduced. So this is part of the work that we've been doing recently. What we did was proving that there's no way to explain this correlation unless you introduce interactions, actual interactions between the species. We did that through a Lotka-Volterra model, so we just add to the kind of model that he introduced interactions between species in the Lotka-Volterra style. And well, the sign of the interaction and the value of the parameters can run over all possible interactions that you find between species. And what we found is that through a Bayesian approach, we were able to find, let's say, a collectivity or an ensemble of interactions that are able to explain the correlation, the kind of correlations that we observe in the data. So here you see, for instance, the dots are the empirical data, and the gray is the curve that we obtain through this Bayesian approach, right? And you can see that we can fit pretty well the correlations in many different ecosystems. The conclusion we obtained is that you need interactions, but the kind of interactions you need are essentially sparse, very sparse, meaning that the interactions that are commensalistic or amensalistic are prevalent. And this is an interesting conclusion. If you want to know more about this, I'll be happy to tell you more. Okay, then I move to the next topic. The next topic is the genotype-phenotype maps. Well, I don't think I have to explain what the genotype and phenotype are in this context, right? But the interesting thing about this is, well, I'm not going to explain you how the flamingo emerges, okay, from the DNA. So I'm going to be more modest and talk about molecular evolution. In molecules, for instance, the RNA, which is a benchmark for this kind of studies, you have the secondary structure. The secondary structure, as you know, is you have the sequence of letters that defines the RNA, and it folds because there are hydrogen bonds interacting between the pairs of letters, and they form a structure like this, which is called a secondary structure. This is the phenotype. That one, the sequence of letters, is the genotype, right? The interesting thing about this is that when you have the phenotype, there are mutations. You can see, well, I hope you can see here that there are two mutations, but these mutations do not change the structure. So there are mutations, meaning that you have different genotypes, and nevertheless they have the same secondary structure. So there is a degeneracy in going from genotype to phenotype. Actually, this degeneracy is very large, because if you make a combinatorial estimation of how many secondary structures you can find for a sequence of a given length L, you find that this is an exponentially increasing number. The number of genotypes is also an exponentially increasing number because there are 4 letters, so it's 4 to the L, and then you divide one by the other and you get the mean number of genotypes per phenotype, and you can see that this is something that is also exponentially increasing. So there are a huge number of genotypes all folding in the same secondary structure. Okay? Actually, this number is not at all representative because it turns out that the distribution, if you compute the distribution of the number of phenotypes having a given number of genotypes folding into that, which is what I'm going to refer as the size of the phenotype, well, the distribution is very uneven and it's actually long tail. This is empirically what you see when you fold molecules of length 20, right? You can see that this is roughly well fitted by a log normal distribution. Okay, the log normal distribution covers, as you can see, a very large range of sizes. So you have phenotypes for which the only 100 or tens or hundreds of genotypes fall into it, but you also have here phenotypes which contain of 10 to the 10 number of genotypes folding into it, right? So there is a huge span of ranges in the sizes. Okay, part of what we did was devising a mathematical model that actually proves that this is the kind of distribution that you do find in this model and in other similar models of the map of genotype to phenotype. So there is actually a log normal distribution, and the two interesting things about this is, first, the mean of this distribution increases with L, which is expected, right? But also the variance increases with L, meaning that if you have very large molecules, the span of sizes that you have increases a lot, right? That's very interesting and has an important consequence, which I'm going to try to explain here. The orange curve is the distribution of phenotypes, right? So this is the number of phenotypes that fall, well, that contain a size or are of a given size, right? And the span of the X is very large, as I said, right? But if you want to compute what is the probability that if you pick a genotype at random falls into a phenotype of a given size, you have to multiply this by S. And that's in the case of log normals has the effect that it shifts the distribution, right, to the right, like this. More so the longer the sequence. So the implications of this is, if you look at, I don't know, here's 99%, but it could be 99.9 or 99.99% of the genotypes that you can pick at random. So 99.99% of them fall into phenotypes that are in this top part of the distribution. So that means most likely evolution is unable to find small phenotypes, even if they are better or fitter than phenotypes that are more abundant. And actually that's what you find. When you go to the database, what you see is that the molecules that you observe in nature belong to the abundant types. And you never find something that has a very low abundance, right, in terms of genotypes. So even if the optimal phenotype is here, you will never find it. So evolution will never find it. Nevertheless, you can find within this region phenotypes which are as fit, almost as fit, as the fittest one that you would find in the low abundant ones. So that's a very interesting implication of this structure. And finally, and I'm going to talk a little bit more about this, is the gene genome networks. The inspiration for this work was this work here, this publication, in which people studied, the authors studied what is the, well, they tried to study the virusphere, right, and the connections, the genetic connections between the viruses in the virusphere. And what they did was drawing a bipartite network in which you have here in color dots, you have the different genomes, meaning different viruses or species of viruses if you want. And then in small dots, you have the genes that you identify in those genomes, right? And there is a link whenever a gene belongs to some genome, okay? Well, they did community analysis, and they were able to classify the viruses in large groups with more or less fit the classes, the taxonomical classes in which you classify viruses. But the thing I want to, or that attracted my interest, was the distribution, the degree distribution that you observe in these networks. What you see is that on the left you can see the degree distribution of the genes, and you can see that it follows the power law with the k minus 2. The exponent is minus 2. But on the other hand, if you do the same thing for the genomes, what you see is something that looks like flat, but it's actually an exponentially decaying distribution. So you have two different patterns when you study the degree distribution of the two things, objects that you are relating there, the genes and the genomes. And we have devised a mechanistic model that more or less reproduces these patterns. And the construction of the mechanistic model is very simple. What you do is, the main mechanism is horizontal gene transfer, which means on the left you have the genes, the green ones. On the right you have the genomes, these are the orange ones. The links are, there's a link whether a gene is found in a given genome, and what happens is some of the genes that are present in a given genome move to another genome in which they are not present, and they form a new link, the red one over there. Of course, this happens with a probability which is proportional to the degree of the gene, because that means the higher the degree, the more genomes this gene is present in, right? So it increases the probability that the gene jumps to another one. Then there is the arrival of novelty, which is a new gene coming from I don't know where, incorporates to the existing genomes, and it introduces a new gene, or an existing gene, the third mechanism, an existing gene founders a new genome, right? Or both happen at the same time. There is an arrival of the new gene founding a new genome, and finally there is gene deletion. I mean, some gene disappears from one of the existing genomes. So these are the basic mechanisms, very simple mechanisms, each one with a given probability. And then when you put these mechanisms into work, what you find is that indeed you observe a degree distribution for genes, which is a power law. You reproduce the power law by fitting properly the parameters, and you also reproduce the exponential decay of the degree of the genomes, right? And this is actually the connection that brought Cecilia to invite me here, I think, because I asked her just to know whether this mechanism is more general than just gene on genomes in viruses. I asked her whether there is a database of hosts and parasites, and the same kind of mechanisms can explain the distribution of hosts and parasites in the bipartite networks. So actually this is the reason why I am here, I guess. Okay, and with this I finished. Thank you very much.
Well, very nice talk, Jose, as usual. I have some questions of the first parts, but I want to focus on the last one, because you said these are the basic mechanisms. You saw in simulations that if you delete one of these mechanisms, you don't obtain the results. Why B plus C is a basic mechanism and not A, B plus C is another basic mechanism? So this...
Well, actually, B plus C is like, well, it's obvious, right? I mean, if you have these two, why not having this third, right? Yes, we...
Why not other? That's the question.
Yeah, we tried different combinations, and we tried new ones, and we tried to delete some of these, and it doesn't work.
Okay.
So anytime you do that, you get a distribution that is not this one. So this seems to be the basic mechanism. Clearly, this should be here, because as you know, this is the basic mechanism to produce this power law. The moving of a gene to a new genome is proportional to
the degree, yeah.
To the degree, and that is
But the key is to reproduce as well an exponential decay
exactly,
for the.
Exactly, and for this
This is the minimal one.
Yeah, these are the minimal. Actually, I must say that this has to be very small.
Mm-hmm.
This has to be extremely small, right? For the reproduce. If this is very large, then it breaks down. And we have tried other mechanisms like duplications of genomes, etc., like trying to reproduce all the possible mechanisms in evolution, and it doesn't work.
And the fitting parameters were not.
Were too small.
Dependency.
If you want to fit the
Okay.
actual,
Okay.
So they are basically
The ones.
non-existent.
Okay. Thank you.
Do you think it's possible? Well, thank you, Jose. Do you think it's possible to think about this model? Think about species interacting and not genome, like parasite and host species interacting, and these mechanisms could be used to also simulate spillover?
Yeah, that's my hope. Actually, yes, I think so. I think these mechanisms or small variations of them should be able to explain exactly the same pattern. So that's why I'm interested in those data, to see if they fit also these distributions, right? And I can explain it with the same mechanisms. Yeah.
Okay, anyone?
Just a little question about that. So the appearance of new genes or genomes are related with those that are already there? I mean, it's like a kind of mutation or something like this, or it's random?
Novelty, I mean, they come from outer space. If you want. I haven't figured out a mechanism to explain why they come here, why they are new genes. But you can imagine, for instance, that there's a meta community, or there are patches and it comes from a different patch where it is present but it's not present here, something like that. Or actually, figuring out the meta version, so to say, of this mechanism is very easy, right? You can imagine this in different patches, and then you introduce these alpha and beta mechanisms, or actually alpha and beta, B plus C, from the amount of or the abundance of these genes in different communities, right? So yes, exactly.
Thank you, Jose.
Thank you.
So our next lecturer will be Dr. Sabrina Araújo. Sabrina is Associate Professor at the Physics Department at the Federal University. I'm not sure why we are having this kind of... So we have a little bit of yeah. So anyway, I'll just introduce Sabrina and I'll see what's happening. So Sabrina is Associate Professor at the Physics Department at the Federal University of Paraná, Brazil. And right now, currently, and she's currently a visiting researcher at the City University of New York, USA. In Brazil, she runs the Lab of Ecology and Evolution of Interactions. Her current work involves mathematical models to investigate species interaction, adaptations, and evolution. Sabrina?
Well, thank you for the nice introduction. Thank you, Cecilia, for the invitation, Ana Paula. It's a pleasure to be here. So, just 15 talk, I'll go through this kind of my research topic. And so, as you said, I'm from Brazil, from Universidade Federal do Paraná, south of Brazil. And the idea is to model animal behavior, ecology, evolution. It's my field work, and for sure I'm not able to talk about all those things. I will be more talk more about host parasite evolution and the ecology, the interaction, and also the evolution. So what I do is model, and just to start to say that when you model, we need to simplify. Yeah, so all models assume simplifications, and we have to have this idea when interpret our data, and simplification is important, but we need to have this in mind that we are doing simplification of nature. And to thinking about this true trophical level, like host and parasite, the tool that I use to play is considering individual-based models, and individuals can be characterized by a phenotype, by a genome, and here, for example, each dot, each square, it's one individual, and the color could mean a phenotype. And we can make question, for example, how will the phenotype evolve when we put these individuals interacting? As Cecilia just thought in the beginning, what's similar to her. So to model that, we need to put some events occurring, so individuals can interact. It can have a fitness associated with this interaction, and then we have new generation when genetic novelties, evolutionary novelties can emerge, new phenotypes can emerge, and then we can end with modeling evolution and ecology. As I said, we need to impose simplification, and the usual thing to model interaction is assume the individuals interact randomly. So we'll select one consumer, we interact with a resource, and usually what the models do is, okay, let's start randomly, and there is a probability of this interaction occur. But for some animals, this is not real, and we can make question if this animal behavior in the sense of the pharyngeal way, or for the avian brood parasite, we need to choose a nest to their leg. So they not try to put a leg by random. They can decide where to interact. And what we made is make this question: is this complication in a model would change the dynamic? It would change our conclusion. So we compare the simple model of consumer and resource, and assuming that and compare when the interaction is random to the situation where the consumer knows a subset of the individuals and can choose, oh, this one I would follow this, try to interact with a given resource. And in this first study, I need to thank Minos Van Balen, where I did one of my postdocs. And the idea, okay, besides of the interaction, we also need to assume the fitness. And here I will show a situation one for the consumer. Okay, for who is online, maybe it's better to use this. So it's the fitness for the consumer, assuming matching. So it seems that for the consumer, it's better to have a phenotype that matching to the research. And there is also external pressure, as Cecilia just explained. So I will save a little bit of time. So this external pressure, you just bound the phenotype to not evolve to so extreme values. And for the resource, the fitness decrease here when the phenotypes match, because it's an antagonist interaction. So we could expect that the evolution will drive thinking about this pressure on the resource for different, so they need to escape, yeah, from the parasite phenotype. And so this is the idea. So choose, compare random, and then we have these two kind of interaction pressure. And here the kind of results that we have when we have this preferential or random. So just explain this graph. Here we have time, and here's the phenotype for the resource, the phenotype for the consumer, and here the population over time. Here the same, this number here, just the strength of the interaction pressure. So I'm increasing here. So what you have here, we have basically similar in the way that is the phenotype can evolve, can oscillate, but I would like to highlight here the strength to this extreme phenotype here that go more extreme here than here. So it's not the same scale. So for when we have this preferential strategy, we can end up with a more extreme phenotypes. And we did all, Sabrina, maybe it's just because we don't impose more strength here. Oh, sorry. Strength here, keep increase this alpha value, but we did that and we never went up with so extreme phenotype. Because here, if the animal do not interact, the consumer do not interact, they will die. So we never end up with so extreme phenotype. So we saw that this preferential strategy can lead to more extreme phenotypes is one of our results. And another thing is this kind of pattern here that just emerged when we have this preferential strategy. And this pattern is that, okay, as we have the matching, we could expect both evolution towards each one of the minimum, so negative or positive, there is no difference in the model. So sometimes I run this, go to the negative. Other runs go to the positive. But we always have something happen, they stay stable in one of those in each simulation. So it's like the consumer is locking this phenotype here. They go and lock the phenotype here. And when the interaction is random, we never went up with this kind of pattern. Actually in this study, we made some analytical studies on the equations. We could see that it's true that it happened, that it's associated with that. But I will not go in detail on that. I just skip this part, okay? So we can talk later on how we could do this analytically. But as Cecilia did in her study, we also make this same question, but in a community level, when we have many results for many consumers. And here, all those resources are resources for any one of those consumers. Okay, they could interact. And our question is, beside of the phenotype evolution, how the network emerged. So we do not impose who will interact with whom. And our question is to see those network structure and the phenotype. And the same question. And the same compare with preferential or random. And we have the external pressure. We could, for example, here in this example here, we could impose the same external pressure for all phenotype. And then we could expect that end up that, okay, everybody will interact with everybody because they are equivalent. And another extreme, oh no, let's put different optimum phenotype, the external pressure put different, and we could expect some result like this. So they have similar optimum, they will co-evolve and stay just one more direct specialized interaction. And I need to thank Alan Avorenga, who was my PhD student who developed this study. So So what you saw, regardless of one of those options here, we have the same qualitative results. So I will not show the difference results between these two, but I can say to you that our result does not depend so much on this kind of thing. For sure, there is a limit for that, okay? But in general, and comparing preferential and random, we saw again that we have more here, have the same scale, so it's easier to compare now. And we see that it's for some simulation, this time again, we have this extreme phenotype again occurring. And here, when we assume random, this extreme phenotype never occur. Here the optimum imposed by the external pressure, it's random, so it's basically when the simulation starts. So they start around and stay around the optimum. Another thing here is just showing the average. Each line is one species, one group here, but I'm not showing the variation in the group, just to make the plot cleaner, okay? And blue means resource, and red means consumer. And when you look for the network, we here the frequency of interaction between the last simulation, basically the last 4,000 simulation. Oh, sorry. So it's not just one time, but it's for the last time here simulation. And when we have this preferential strategy, we saw some, could say, coevolutionary units when group of individuals. So here's the resource, and here's the, oh sorry, here's the consumer, and here's are the resource. And color means that those species interact, and light is not so often, and dark is, so the interaction is often. So most of the time this group of species are interacting. So we have this group of individuals that co-evolving. So we call this the coevolutionary units, like Bascomte first introduced this name. And when we assume more random, we also end up with a kind of pattern more random. So there is a probability to the interaction to be succeed. So it's not random, okay, the interaction will be random, so your graph will be random. No, we have a probability of the interaction occur. So, but we never end up with so clear coevolutionary units. And here we are not talking about speciation, you know, just to have it clear, just this time scale when we have just phenotype adaptation evolution. I'm also interested in speciation. I still have a little bit of time. Put my clock here. So just to have an idea what I have done about speciation, we did this. I need to thank everybody that is here, especially Elvira was a PhD student of mine. And what it did here, sorry, I'll explain here. We use the phylogeny of the host and let the parasite evolve in a given empirical phylogeny. And here we do not assume coevolution in the sense that the parasite imposes selection pressure on the host. So here we assume that the hosts are evolving for another reasons, and the parasite who needs a resource are evolving the resource. So it's more evolution of the parasite guided by the host phylogeny. So each individual parasite, we model that, and we can now we model the genome and not the phenotype. So I'm not talking more about the phenotype, we model the genome. And we have strategies, and I'll talk in my another talk in Wednesday how we do that. But we can measure speciation and recognize when it happened. And we compare our data with 9 empirical data that, I'm sorry, I'm a physicist, I don't have, so it's Elvira who knows each one of those. I have an idea about this there, but I want to show you. So now we can build the network that is evolving. So here's the phylogeny of the host, and it is empirical data, so we're not modeling that part. And here's the parasite phylogeny that we are modeling. So we are modeling this using this host through time, through the ecological time of the host. And we do this through time and compare to the empirical data. So here's time going on, the empirical data is just in the present data. And as you see here in my simulation, we have more links. We also have more species of parasite. That maybe is true. We do not know all parasite species. So empirical data is just a sample of what is going on in nature. But we end up with a kind of similar pattern. We measure those patterns, and and of course we have parameters in the model. And one of important parameters is the intensity of host switching. How often will allow an individual to try to use another host? One parasite individual try to use another host. And comparing those 9 is my last slide. Okay, so I'm finishing. And we compare which intensity of host switch better fit the empirical data. And we found that for some empirical data, low intensity of host switch fits better, and for another empirical data, high intensity of host switching fits better. And those data seems to be those one that is more in regional scale, that has low intensity of host switching, and those that is more local parameters of high intensity of host switching fits better. And it seems that makes sense in the way that if they are local, they have more opportunity to host switch. So maybe what's here in my simulation, what's driving the generalist is more opportunity to interact. So it could be something that emergent from here. So that's it. Thank you. Just to summarize, models assume simplification, and we need to have this in mind, but it brings insights in theory. The no-random interaction can play a relevant role in evolution, and we can use these tools to study species evolution, speciation that I just showed you, but we can also study extinctions, yeah, because we are modeling, you see the extinctions. Thank you again. Mm-hmm.
So we talk, I mean, as far as I understand, this is part of the question, well, you study this, well, interaction also at the community level, and well, in this case, as far as I understand, well, you detect this modular structure, this, well, community structure. So you say that the species are organized in groups. I mean, my question is the following: do you also find a structure within the blocks within the group? Do you study this? Something like as any block nested, something in this direction. So did you study?
Yeah, we didn't look at that. We thought, yeah, it would be something interesting to look at, but we didn't.
Okay. Well,
yeah.
Thank you.
Okay, great talk, Sabrina. I really like your last work. It's amazing. But I'm kidding. So when you say, I think this is good. When you say preferential and random interactions, could we say that when you have the preferential one, you have at least one direction for the interaction, at least for each individual?
Yeah, for each individual, yes, because if the individual already carrying a phenotype, so I don't know if I don't think it's directional. Because the individual have a phenotype 5, it prefer phenotypes around 5. It could be 5 plus something or 5 minus something. So in the point of view of the individual, also there is not a preferential direction, but it's around 5. So maybe 5 is already directional in some way.
Yeah, compared to the random, there is a direction, no?
There is a direction. I don't know if I understand the way direction, the sense going to the direction of a greater phenotypes than a smaller phenotype.
Yeah, going direction to a phenotype.
Okay, yeah.
So there is.
Yeah. So yes, there is a preference in direction to a given type of phenotype. Yeah, because when you said direction, I thought about this kind of direction here. So it's not.
This is a consequence, I guess, maybe.
Yeah.
No?
It's not so obvious that.
Okay.
Yeah. So here we have. Here's the host phenotype that is below. No, this is below the consumer phenotype. So what happened if a phenotype evolved from here to a little bit greater value? It would be preferred, and then the consumer catch it, and this phenotype cannot evolve. And if the interaction is random, there is a greater chance of it escape. So it's something that is what is going on and why it's locked in just one direction. But but it could be, I don't know.
Yeah, I was just trying to let you know what I was thinking. That for me the preferential one sounds like natural selection, and the random one sounds like genetic drift, for instance. So one is being locked in some place, and the other one is drifting and trying new configurations.
Yeah, but it can also have this
pattern in the other one.
The other one. So
yes.
It's not so obvious.
So obvious.
There will be a lock.
Okay, thank you.
Thank you.
Just one question. Is that it? Is that okay?
Thank you. No, I'm trying to make like a parallel with the modeling framework that I was working with, because you use individual-based models and I was using like mean field. And I don't know if I understood what is preferential attachment and what is random, because in both cases you have selection to trait matching, no?
Yeah.
So what does it mean, the preferential?
Okay. So it means, for example, let's assume that consumer individuals can have in his neighborhood, interaction neighborhood, 5 individuals. And in the random, just pick one of the 5 randomly. And the preferential will look among those 5, okay, this one will increase my fit, so I will interact with that one. Okay. And this size of the neighborhood is something that we played. Okay, if it know the whole community or just a part of the community, how it will change. So it's something that we played with that. Of course, if choose among one, you have the random version. Yeah, so that's the way that we got the random version. So we have just one option, it's just random. We have 2 options, and then we compare them. Flavia knows how.
Thank you, Sabrina. So now I would like to introduce you to Flavia Marchitti. Flavia is Associate Professor in the Department of Genetics, Evolution, Microbiology and Immunology at the State University of Campinas, Brazil, and the Scientific Coordinator of the Training Program on Quantitative Biology and Ecology from Serra Pilheira Institute. She is mainly interested in the application of mathematics in ecological and evolutionary complex systems. So, thank you.
Thank you for the introduction. Can I move this to here? Because I need to be close to my screen. Is it fine? Can people use this one and turn that one off? Okay. Can you hear me? Yeah. I guess you can, right? Okay. So I will be talking about signatures of microevolutionary and ecological process in phylogenetic trees. I think it's a little bit different of what has been talking before, but I think there are many links there, and that's why I'm interested in many stuff. First of all, I would like to thank all the people that helped me with this work, and also the funding agencies and the institutions that I've been in the last years. I don't know where is the arrow here. So I think I'm very interested in diversification, but not only. And I think many people in biology, in evolution, they are very interested in how species diversity is possible, how new species appear, how these species establish, and how they interact with each other. So I'm very interested in species diversity, but also I would like to understand, for instance, the evolutionary patterns. This can be represented, for instance, by phylogenetic trees, how these species are related, and maybe how they emerged from another species or from common species. And I also like to understand what are the shaping processes that are generating these patterns. For instance, the space can be an important component on forming new species. And something that we call in this area is that we have macroevolutionary patterns and microevolutionary forces, and here I'm doing the Brazilian accent just to understand what I'm talking about. So the macroevolutionary patterns and the microevolutionary forces, when they are together, we call this the micro-macro evolution. But also there are ecological processes that maybe can shape the evolutionary patterns. And for instance, the geographic configuration is a very known process that can shape how species emerge. And this, the allopatric speciation is very studied because it was very important, for instance, to some species like these apes to emerge. And so this is only when a barrier appears and these individuals are separated for some time, and they are recognized as different species. But also sympatric speciation, when these individuals or these organisms are in the same space, but they are actually different species, even sharing a common space. So how is this possible? How this, for instance, these cichlid fishes, they appear just sharing a common space, common resources and everything. So one of the things that I want to know is that so we have these two different processes. So I forget the allopatric speciation here. Does it leave any signature in the phylogenetic tree? Can we detect from the phylogenetic tree what was the process that gave rise to this species? So I've been using, one of the models that I've been using is the Derrida-Higgs model. So this model, as Sabrina told a little bit there, it's a genome explicit model, and I've been working with an individual-based model approach. So we have individuals. Each individual has its own string of genome, and then they can reproduce sexually. A focal individual finds a sexual partner, and then with the distance, a given distance, if it's lower than a given value, they will form an offspring that can mutate, but every position here in this string that I can call alleles, they come from the focal or from the sexual partner with equal probability. And if I'm interested, for instance, in this space, I can put these individuals in the space, right? It's easy, I just put them there. It's an individual-based model. And then I can find that over time, groups of species will share some genetic flow, but some of them will break the flow. They cannot reproduce, the individuals of this group, they cannot reproduce with the individuals of the yellow group, and here the blue, yellow, and gray cannot reproduce with each other, so just inside. And then I have what I call species that are just the individuals that are linked by the genetic flow. And with time, what we observe in this model is that group of species or group of individuals that share a genetic flow, they form in this spatial configuration. And as I told you, if I'm interested in a spatial configuration, I can use, for instance, a radius as that defines the radius where an individual will find its sexual partner. So this is just the method I've been using. This radius can be small, this radius can be large. If it's small, we call parapatric. If it's large, we call more close to a sympatric situation. And what we observed is that over time, with this model, the number of species, depending on the radius, can change. So I will focus here just in the blue, yellow, and green. So what we observed is that when the radius is smaller, we have more species forming. When the radius is larger, we have less species forming. So in symmetric situations, what we see is that we have less species. And we had to develop, to understand better what are the phylogenetic signals in these situations, we had to develop a method to construct the phylogenetic trees. I will not enter in detail in this work, but it was something that we had to develop. And just put in these results so we could see the sympatric speciation and the allopatric and parapatric situation evolving. I would like to see what were the different phylogenetic trees that were forming. And then here I have two structures of the phylogenetic tree. The balancing of the phylogenetic tree, and here is the acceleration of the branches of this phylogenetic tree. And something that we see is that, so we have a range here of how balanced this phylogenetic can be and how tippy or how steamy this phylogenetic tree can be. And what we find is that, again, looking at the blue and yellow and green, oh my God, I don't know the colors anymore. So looking at these three colors, what we saw is that the radius here makes a difference on the branches of this phylogenetic tree. So as you have a larger radius or a more sympatric situation, you have a more tippy kind of phylogenetic tree. And then I will not enter in this context here, but we had different real phylogenetic trees here. They are adaptive and adaptive, and they fit more or less with the situation of how the genetic flow was during the speciation of these groups. But then, just focus again on the blue, yellow, and green. As we decrease the radius, or as we made the spatial configuration more structured, we have like more small balls inside that lattice. We saw there was a difference on the branch length of these phylogenetic trees. So I thought, oh, maybe if we have a more structured spatial configuration like this one, we have like beams or islands, whatever you want to talk about, or more, it's a more allopatric situation, I would expect that still I would have a signal on this branch length of my phylogenetic trees. And something that I could observe is that actually the branch, they didn't change a little, they changed a little bit depending on the number of species, but just on the spread of these points. But what we observed a lot is that actually the number of beams or the number of islands that we had would make a great difference on the balancing of these phylogenetic trees. The balance of phylogenetic trees is dependent of the number of species. It's something that we cannot control. But what we observe is that those species that emerge in a, for instance, 4D situation or for island situations, they have a lower balancing on their, they are more balanced in their phylogenetic trees. Okay, and then we could observe as well that in the real data or in the real phylogenetic trees, When I say real, it's not real because they are all approximations, but it's what I call here. On the observed phylogenetic trees, we could say, for instance, for some species here, oh my God, the silver alliance, the tetragnatha spiders, and for the stictalicans, we could say, based on the balancing of their phylogenetic tree, what was the configuration that they emerged from. But we could not say it from, for instance, from the Darwin finches. And maybe because Darwin finches, they fly, and they don't see the environment as we see. Maybe they see just one big island, or many islands as just one space for them. But I cannot say that, right? Okay, I think I don't have time for this part. I will just skip just to go to this other part. And in the recent years, I've been incorporating some kind of selection or some kind of selection in this work, and I've been doing this through a game theory approach. So we have individuals that they have their genomes, and they also have a trait. Here, I've been using a game theory approach in a prisoner's dilemma case. So they are cooperators and defectors. They could be any other, it could be any other strategy. So here I'm using just this one. And in this context, I think it's my phone. So what we saw, what we had before from Novak's work is that the trait of cooperators, they can evolve better in a spatial configuration. And then we wanted to understand how this could be on a scenario where speciation is also happening. We have space, we have these traits of cooperation and defectors, and we would like to understand how this could affect the fact that the species are actually sharing common co-specifics, they are forming species, how this could affect the speciation process. And what we observed is that cooperation, the richness of the species didn't change a lot with game and without game, so we have here two situations. The number of cooperators changed a little bit depending on the values of the benefit for the cooperator. But something that we observe is also that the phylogenetic trees were very different. Without game and with game, so selection, in our case, it was making a difference on the shape of this phylogenetic tree. So not only space, but also selection seems to affect a little bit the shape of these phylogenetic trees. And currently, what we want to understand in this context is that if cooperator species, the species that form that are actually cooperator, if they vanish more frequently, and also if hybridization could mix species with different strategies. If we could mix species, form species that are more cooperator, species that are more defector, if they mix in hybridizations. And also, something that I'm starting now is to understand, for instance, how coevolution, in the sense of cophylogenies, could emerge in situations like plant-animals interactions and how parasites interactions under different spatial configurations. And to understand how what are the signals in the phylogenetic trees, where they are coming from. If they are coming from the coevolution with the other species, if they are coming from the spatial configuration, can we detect where from where these patterns are emerging? I think that's it. I'm in time, and I have time for questions. Okay.
Hi, nice talk, as always. I have a question about the game part, obviously. So, how you do this thing about the evolutionary part when there is this game involved? I mean, because as far as I understood, you have like two mechanisms, right? You have one that is related with the reproduction, you know, like the ones and the zeros and this common stuff, and but in the other side, on the other hand, you have also the cooperator defectors that I guess they have also some impact in the way they reproduce. I guess if they accumulate more more fitness or whatever, right? How you couple these two things?
Is it working? Yeah. Okay, great. So it's kind of similar what Novak did in his work, so I use his plot here to explain better. So you have a focal individual, and this focal individual will choose someone to reproduce with. And when you choose someone to reproduce with, when we do this in our work, we are considering both the fitness and if they are from the same species.
How do you weight each one?
Yeah, so I explain. So the focal individual, we compute the fitness of this individual based on the game, just like Novak. And then for the reproduction, we have this focal individual. It's looking for a partner in the neighborhood. So we look for the co-specifics, and then we choose the one that has the higher fitness. With a probability, we get the one with the higher fitness based on the game.
Okay, so you choose based on the game among those that can reproduce.
Yes, yes.
Have the same question as Luis. And now that I understand the mechanism, why you choose the prisoner's dilemma and not a coordination game?
No,
Should be better a coordination if you base the interaction based if you are in the same species or not. I mean, cooperation or defection at the end is a trait, it's a name, no?
Yeah, it's a name. We have cooperators and defectors in the different species that are formed, right?
Mm-hmm.
I don't say that a cooperator in species 1 is different of a cooperator in species 2. But then we chose the prisoner's dilemma just because it was the first game that we tried, but of course we are trying with different games, and I don't have the results here, but if I remember well, there was not a big difference on the phylogenetic trees that emerged.
How do you populate the initial condition when you have like islands? How you populate the population?
So in this case here, right? Or even in an island.
When you have like these teams, islands, or
we start with all individuals. They have the same genome, so they are genetically equal, and mutations appear and make different.
Okay. So everybody starts.
We start and we don't start with species. The species start to form. Okay, thank you.
No. I'm gonna skip it after. No. We go. I am just
because of the time. Okay.
If it's okay for you, it's okay for us. Just pin this. You can put it on there.
So thank you, Flavia, for your great talk. Now we will introduce you to Dr. Jesus Gomez Gardeñez. Jesus is a full professor in condensed matter physics at the University of Zaragoza in Spain and leads the group of theoretical and applied modeling at the Institute of Biocomputation and Physics of Complex Systems. His research interests involve complex systems and networks, epidemic spreading, emergency of behaviors and cultures in social systems, and synchronization phenomena. Thank you, Jesus.
Thank you. Thank you very much. So I'm really happy. I'm sorry, I'm physicist, so I think this session is, yeah, I know. I mean, mathematician, physicist, again, mathematician. So I'm sorry. I'm very happy to be here. Thank you for the invitation. Yes, I mean, after the introduction, I don't think I have to explain that in my lab what we do is to try to explain how this collective phenomena that can be very different emerge in large ensemble of interacting units. I mean, can be humans, can be computers, can be neurons, can be animals, can be viruses, etc. So here I will focus in this specific line of research, which has been probably the most important line of research in the last years because obvious reasons. And yes, I'm a physicist, and as I said, I come from this statistical physics view, in which we try to connect what we see at the macro level, at the community level, if you want, with what's happening in the interacting the level, okay, what we call the microscopic world, okay. This is something very important in physics, as you may know, and after many years and decades of development, some physicists, Nobel Prize physicists arrived. It was Philip Warren Anderson that claimed, and he was very angry with high energy physicists that have all the money for them. So say, okay, it's very nice to break the nature into pieces and to know what is going on at the nanoscopic level, if you want. But there is another view which is as important as the reductionist one, which is the one of trying to build systems and try to understand how complexity, how macroscopic properties emerge from the interaction of many particles, in the sense that you cannot explain what's going on at the macro level by just looking a particle. You cannot explain what's going on in the brain by looking at the neuron. You cannot explain what's going on in an economic crisis by looking just a bill of money, okay? So this was the perspective, and that's why we study collective properties, synchronization, cooperation, social behavior, and epidemics, okay? And epidemics, what we try is to understand why some localized outbreak in a place can yield a big epidemic wave, or even a pandemic. In the last years, there have been, I'm sorry, I mean, we are full of movies, but in PDF it was impossible. So it was a large movement towards what we called epidemic forecasting, and is what we want as physicists, and not only physicists, of course, but also mathematicians and epidemiologists, is try to be as good as weather forecasting. And remember that weather forecasting emerged in 1922, which is the same year as the very first compartment models in epidemics appear, okay? Obviously, as you may see in your mobile phone, the development of weather forecasting has been much better than epidemic forecasting, but it all started with a set of equations, which are a set of differential equations involving thermodynamics and hydrodynamics, etc. And one of the creators of the field, Lewis Fry Richardson, said that perhaps someday in the dim future, at that time obviously, it will be possible to advance the computation faster than the weather advances, and at a cost less than the saving to mankind due to the information gained. But that's a dream. That was a dream. Now today you can have the forecast in your mobile phone. So this was how Richardson thought how a factory of weather forecasting should be. Many people, I think he calculated around I think several thousand people making calculations obviously, simultaneously with all the data from the world. Nowadays we have supercomputers, we have high resolution data, and all together, and obviously more refined models, make that we can forecast weather, and even more, we can forecast extreme events, which is very difficult, okay, because of the high nonlinearity. So the question is, if in my field is if we can do the same thing with epidemic models and try to make epidemic forecast systems. There are many questions, but we have to start with the beginning of this field. I know that most of you know what is a compartmental model. I mean, this is the basic piece of a epidemic model in which you have a population divided in compartments. Here is a very simple SIR, susceptible infectious recovered model, in which you have people in each of these three compartments. They can make transitions according to some parameters. Here only lambda and mu, which are the rates of infection and recovery. And the important thing here is that you have to compute the basic reproductive number. Okay, this is you make a model, you make the calculations. Typically you assume you are in a mean field population, so everybody interact with everybody with the same intensity, and then you derive mathematically, if you can, the basic reproductive number. The basic reproductive number, that, as you know, is the average number of infections that a single infectious person produces in a population of fully susceptible people. Okay, and according to physics, okay, there is a phase transition. Okay, according to mathematicians, there is a bifurcation in which you have a critical number for this parameter, for this quantity, better said, which is 1. Okay, so if R0 is less than 1, you don't have any epidemic because the average number of infections is less than 1. If R0 is larger than 1, then you have an epidemic. Okay. The good thing is that R0 can be measured experimentally from data, okay? Especially when you have an outbreak, you assume there is an exponential evolution, and then you obtain from data this quantity. And then you can check models, because each model gives you a different expression, okay? These are very simple models, and as you see, after some mathematics, you have different expression for R0. At the end, regardless of the model you use, the R0 is just the product of 3 factors, okay, which is the number of contacts you made per unit time, the probability of infecting a contact, and the time you are infecting. So the time window you are infectious, okay? And obviously, depending on the model, these 3 quantities, T, C, and P, have different values. And the interesting thing of this, and this is the basic building block of mathematical epidemiology, you find books and books and books about this, is how to calculate for each model these quantities, T, C, and P, because if you know this, then you can act into the system. Because, for instance, if you reduce C in order to reduce R0, because you are isolating people, you are reducing contacts. If you reduce P, it's because you are reducing transmissibility, because you use, for instance, mask, or you use vaccines. And if you want to reduce T, then you want to isolate people. Okay, so for instance, making tracing, contact tracing after the detection. So it's easy to see the influence of each factor in each disease mathematically. There are main limitations, obviously. For instance, is that all societies are identical, and here you have real estimations about the first COVID-19 outbreak, and as you see in Europe, only in Europe, the values computed for R0 are totally different, okay? That's why societies are not similar. I mean, yeah, we are not mean field, as you know. Other limitations is, for instance, that in a society all individuals are identical, and this is not true. I mean, there are a lot of heterogeneities. We live in a network, for instance, if we are thinking in a big social network, and in this network you have to compute what's called the epidemic threshold. The epidemic threshold is the translation of R0 in the mean field to the field of networks, which is my field, in which we want to compute what is the minimal infectivity lambda that a pathogen should have in order to trigger an epidemic. Okay, so this epidemic threshold is related not only to the biological quantities of the pathogen, but also on the way we interact. In fact, you can compute it, you can derive it, and you obtain that this epidemic threshold, this minimum infectivity that you need to trigger an epidemic, is related with the maximum eigenvalue of a matrix A, which is the matrix that encodes who interact with whom, which is called the adjacency matrix. It's zeros and ones, okay? If I interact with Cecilia, I have a 1 in our link. As you know, it's really simple. The good thing is that we can also relate these maximum eigenvalues with 2 descriptors that are very easy in networks, which are the first and the second moments of the degree distribution. The degree distribution, you have it there. I think I can, no. You have it here, okay, is the probability that you find a node, an agent with connectivity k, okay? This is the typical signature of a degree distribution in a random graph, in which not all the people have the same degree, but most of the degrees are located around a mean. Okay, this is typically a Gaussian, Lorentzian, as you want. But in reality, what we have is this kind of trend, which is a power law distribution. There is a very slow decay, not exponential at all, but potential. And this means that there are actors like this one in this network, which are the so-called hubs, that are really well connected to other people, with a degree which is orders of magnitude larger than the average degree of the population. And this is very important because in these networks, in real networks, this quantity here in the denominator is really large. And if it is really large, it means that the epidemic threshold is really small, much smaller than expected. Okay, so the problem is that if you have two networks with the very same degree, average degree, sorry, but corresponding to two different degree distributions, a Poissonian one and a power law one, you have different behaviors for a pathogen that in principle should not trigger an epidemic. In the Erdos range one, in the random graph, sorry, you have this phase diagram in which you have that there are no epidemics. You are lying in this area in which no epidemics is found. But if you are with the very same pathogen, with the very same infectivity in a network with its degree distribution as power law, then you will have this curve that means that you find an epidemic state. Okay, so two different evolutions as you have here for the same pathogen, here for a random graph and here for another graph with the same average degree but with a degree distribution which is power law. Okay, so this was the end of R0 as we knew it, okay, because in principle this pathogen should not trigger an epidemic in the mean field context, but the problem is that not only is the infectivity, the intrinsic infectivity that you find in the lab, but it's also how we mix. Okay. Another, well, this was very important. I mean, this was very important because we know that as humans we are not encoded in a fixed network, but there are other items that are encoded in fixed networks which are highly heterogeneous, and these are computers. Okay, computers, and this phenomenon that I show you explain this strange case, the strange case of the virus I love you. The virus I love you was not a virus. I mean, was just a worm encoded by a student of computer science, a student in Taiwan, no, Manila, sorry, in order to steal the Wi-Fi password of the neighbors. Okay, he's in jail. He's still in jail because it caused billions of dollars of damage. Okay, and still 20 years after this happened, this virus is still running in the network. And this is because the network is a scale-free network, and even a pathogen which is seemingly inoffensive, it causes damage which is really large. Okay, this is a field, network epidemiology, my field, that started with this the first data about interactions, especially in computer science, and you see that it has a growing trend, especially obviously after COVID-19. You have this huge jump, but it was already in a very good shape, very well before the COVID-19 pandemics. And the next step is to abandon networks and incorporate that we are not living in a closed and independent population, and to incorporate mobility. Why mobility? Okay, mobility is important and was important in order to explain how infectious disease, how communicable disease is spread around the world for ages, obviously. And as you see, you can follow the track of malaria, leprosy, influenza, smallpox, etc., measles also, because we have records, okay, and we can reproduce this track that correspond to human mobility in the ages of when we were hunter-gatherers. Okay, obviously, as we started to construct cities or settlements, this problem started to increase because transmission in community started to increase. The thing is that at that time, these two scales, the scale associated to infection was much faster than the scale associated to human spread, to human diffusion. Okay, it take in this case, this is the bubonic plague in Europe, and as you see, it all started here in the southern part of Europe, and it took several years, 5-6 years, in order to reach the northern part of Europe, okay? Because obviously the time scale associated to the transmission was much faster than our scale to go to another place in mobility. The thing is that in the last decades, these scales has interchanged, and now what we have is a single infection in one place start to spread, and in weeks we have in a worldwide context. This is the case for the SARS from Beijing, the pandemic influenza A in 2009, and obviously COVID-19 virus that after 1 month we have records of infections around the world in all the continents. Okay, another thing which is important to take into account, and as I say, is global urbanization. Okay, our passage from hunter-gatherers to farmers increases the incidence of diseases, and now, I mean, you can translate this transition into the global urbanization transition we are suffering. I mean, in 2009, the global population living in urban environments became larger than the one living in rural populations. And there is some estimation that in 2030, almost 8% of people will be living in megacities, so cities of 10 million of more people, okay? So this mobility and this global urbanization implies a challenge to epidemiology, and this is the challenge that we want to transform into solution, okay? Because urban centers, and also because of big data era, offer incredible opportunities for disease surveillance, control, and preventions that are totally absent in the rural population, okay? And this article claims these new challenges and new problems, but also new solutions that we want to address. And this is something that we can tackle from the perspective of complex systems, because we have to mix human behavior, we have to make epidemic modeling, we have to mix also evolutionary game theory, because also human reaction against measure is important. So a lot of fields coming together, and obviously also ecology, disease ecology, because this is something that is happening together with what happened with vectors, for instance, which is something that we have worked, as I will show tomorrow. So this is my field of research: how to implement how to incorporate fingerprints of human mixing into epidemic models and working to metapopulations. I don't think I have to explain here what is a metapopulation, because this is something that emerged in the field of ecology. But in my case, up to today, a metapopulation was a set of nodes in a network, and the nodes depended on the level of description, could mean urban areas in a city, or city in a region, or regions in a country, or countries in a global world, or all together, depending in the level of clustering. And inside the patches, you find people interacting according to some rules in which they transfer these pathogens among each other, and also moving between patches in order to transfer this communicable disease to longer distances. Well, as far as we have the first data about human mobility, so we have to wait until the beginning of this century, the first epidemic models, the physicists we call reaction diffusion models, okay, started to appear, incorporating metapopulation theory, epidemic models, and real human mobility patterns, okay? In the first papers, of course, the human mobility part was coarse-grained, in the sense that it assumed randomness, it assumed that mobility, the network was not a real one, but progressively we have been able to incorporate realistic pattern of human mixing, okay? And one of them that we in my group have worked a lot in the last years is to incorporate this kind of commuting patterns that occurs mostly in cities. That's why we are very focused on cities, in which we have different ingredients. We have the epidemics, we have also the social segregation, especially in Latin America. We have different ways of moving, and we have also the connection between different cities, especially when they are large, okay? So we have different scales and different spatial scales associated to this problem. This is something that was addressed, as I said, by the community, and in the last years what we have is we have work in this model, which is the movement interaction return. As Jose said, I think I'm here because of this. I mean, Cecilia invited me because of this. This is the contribution, okay? And it's a very simple model, okay? I want to just sketch the simplicity. So you have a population that you divide in different patches. The patches have different populations, okay? So this is the first thing. You can put the data that you have according to the human population in each patch. Then you have mobility. This is something that, I mean, you can construct models of mobility. There is a huge field about mobility models in physics if you want, but you have real data, so you can take real data and construct these kind of models. And then you incorporate also the epidemic model, here the simplest one, which is the SIS. You can be healthy, susceptible, infected or infectious. And then what you have is also a parameter. Obviously you have more than one parameter, which is related to mobility, which is how often people interact in another place different from the residential one, okay? Related to work, related to leisure, related to whatever. And this is the parameter P. And then you have the mobility rules that you have again from data, which is this matrix parameters R that I will explain tomorrow, which are associated to the probability that given that you move, you go to this place. So the probability that if I move today, the probability that I go to university, Complutense University, okay? And then if you do this, you make this interaction happen, you have transfer of pathogens, and again, after the interaction, you have the returning stage in which all the residents to a patch come back to the residential area. And then this is the basic thing. What is the important thing we have done? Okay, what we have done is to use these models in order to answer this question. Given a population in which you have an heterogeneous demographic and mobility patterns encoded by real data, which is encoded by these entries, and I, which is the population at patch I, Ji is the probability that you move from patch J to I. What's the influence of mobility? And the answer is again the inverse of a matrix, which is not now the adjacency matrix, okay? The adjacency matrix was just 1 zeros, if we are related or not. Here is something which we call the mixing matrix, that tells you how people from different patches are related, and depends on demographic features, on mobility features, and also on the degree of mobility. In the simplest form has this form that you have here, okay? In which you have the 3 elementary processes that happen in the mixing of people in this simple metapopulation model. This is the mixing at the residential area. This is the mixing at one residential area when one is visiting you or you are visiting the other, okay? And finally, this third term in which you have the mixing that happens when we both meet in another place which is not our residential area. Example, here today. We are not in our residential area, now we are in a third place, okay? So you can compute, and the thing is that if you have data from a city, you don't have to do simulations. You just take you just take R and N, you construct this mixing matrix, you compute the largest eigenvalue, and then you have this epidemic threshold, okay? And this was the theoretical part. The discovery was something that is common sense. Well, it's not common sense. I mean, if I say to you what happens if we move more, what you say is naively is that it's better for the epidemic to spread, right? I mean, the more we move, the better for the pathogen to spread. No. Okay, depends. It depends. It depends a lot on the structure of the metapopulation. And in fact, here we explore initially we explored 3 different cities in Colombia, Cali, Medellin, and Bogota, and we observed the opposite, okay? Here in Cali, we observed that the more we move, the epidemic threshold started to increase at the beginning. So it means that the more we move, if the epidemic threshold is larger, is that the pathogen spreads worse, okay? This is the so-called epidemic detriment to mobility. But moreover, this was an initial increase, but then you have the common sense, okay? And then if you go to another city, Medellin or Bogota, you have another different behavior in which increasing mobility, the more we move, the worse for the pathogen to spread, always, okay? And, well, this was kind of counterintuitive. Then, if you go to the theoretical model back again, and we will do tomorrow, you understand why. And the reason is because the segregation of people in the residential areas, how people is segregated in a city, is indicative of if you are in a city of type 2, type 3, and type 1, which is the other one, which is the case in which the epidemic threshold is always vanishing, okay? It's always decreasing function of P, okay? So it was a discovery that claimed that in order to make some containment policy in a metapopulation, you have to investigate, you have to concentrate, you have to focus on how mixing happened in this particular metapopulation, in this particular city. And I think I should stop here, because tomorrow we will concentrate on showing some theoretical things about these works, and how we implemented this to stop vector-borne disease, which is, I think, is the thing which is more related to this workshop. So I think I'm on time. Okay. Thank you.
Questions? Thank you, super interesting. I was trying to make some analogies of the meta population model and putting these in a meta community framework. So looking at patches and dispersal among these patches, do you think it's possible? Having what you call what's the name, the migration matrix? No, the
mixing matrix.
The mixing matrix. Can we build this mixing matrix based on the structure of the landscape, like a spatial graph? You know, you have patches of, so that those patches, they are connected by the dispersal of some species that can cross the map, for example. And if we have actual real data, because we have these spatial graphs that are coming from landscape ecology, for example, they work with these kind of spatial networks. So if we have these spatial networks and we have some measures of the connectivity among these patches, can we calculate these? I'm asking this why, because if we are trying also to, this is one of the questions we have, if we want to increase the health of the landscape, let's say, so how can we plan to have those forest patches? Like, do we want to increase the size of the fragments, or do we want to connect those fragments? So because we know that we need to restore biodiversity, so in which configuration should we have for those? I don't know if you understand what I mean.
Yeah.
Like,
it can be also, well, piece by piece.
Yeah.
The quick answer is yes, that's why we are here.
Yeah.
And the thing is that if you have data, I think it's straightforward, in the sense that then we can construct the matrix R, which is the probability that one animal goes to one patch to another. If we don't have data, or the data is scarce, what you can think is about to go to models in which you take into account for instance, barriers, okay, so the landscape, the real landscape, and then construct synthetic models of mobility, okay, in which you can, in principle, incorporate this kind of information about geography, about something that you know from the landscape. So in principle, yes. And yes, I mean, these kind of models, I mean, I have just presented without any equations the basic model, but then you can incorporate a lot of things. I mean, tomorrow I will explain how to use this model in order to allocate resources to of mosquitoes with Wolbachia, for instance. So you can think about this model as a way to design strategies to containment, or to increase the health of an environment, of a landscape. Yeah. That's the focus. Yeah.
Thank you for your talk. It's really nice talk, nice slides.
Thank you. And without movies.
Yeah, I would like to see it. Okay, the model that is behind your network is those classical models, SEER models. Is that what is? Because you do not need to look for the models. You do not do the simulations, just the network structure. Is that right? But you need to assume what is going behind, what would go in behind those traditional interaction models. Okay. Do you have an idea what could change if you play with those equations behind? Any comment about that?
Tomorrow.
Okay.
Yes, I mean the mixing matrix I saw you is just with the SIS model, which is the simplest compartmental model. But if you change the interaction rules, for instance, if you have vector-borne disease or if you have COVID-19, I mean more particular disease, then you have obviously compartmental models that are really not complicated but different, I mean, and more realistic. And this changed the mixing matrix as well. Okay, you have more ingredients in the mixing matrix, because the important thing is not only the interaction, okay, but also how this interaction translates into contagions. And depending on different compartmental models, you have different pathways to do this. So we have played, and in fact is in these two parts. So all these parts theoretical, I mean, is concentrated in how mixing or how to advance in how we capture human mixing. But the last two, the applications, in this case, you change the compartmental model to the Ross McDonald one, for instance, or to the ones in which you have asymptomatic, presymptomatic, I mean, you have exposed compartments. So then you have different mixing matrix, but again, I mean, the strategy is the same as I showed you today. Tomorrow we will see a practical implementation.
Okay. One more question.
This might be actually for tomorrow, I'm not sure, but you need to speak again. So I'm not sure if this is like maybe a better question for tomorrow, but I was wondering if, because you do with metapopulations and dispersal of a single kind of species maybe across space, have you incorporated, or is it possible to incorporate species interactions to go from single species to communities, and for example incorporate the dependence of, say, one virus on a certain host species and where that host species is in space?
That's what we want to do now.
Okay.
Not still. Okay. And the next step, I mean, is this, and also incorporate kind of virus evolution and see what happens. For instance, if you do this viral evolution, as we see, I mean, now Jose is not here, but the time scale is really for virus is really fast. I mean, and we have seen with COVID-19 that you have an evolution that's happening with our reactions. And what we want to do is, in this patchy environment, see what happens if we have different evolutions according to different responses to the virus. For instance, what happened, I mean, is useful if we start vaccinating a set of patches, or we start to concentrate a lot of strict measures, lockdown measures in a set of patches, while other patches, like Brazil, for instance, is not implementing rules. I mean, is what happened. I mean, there is a different speed of evolution. We are restricting the landscape that the virus can explore in different patches differently, and how this impacts of the health in a global level. This is something that we want to do. So different, not only different hosts, but also different pathogens co-evolving. But this is what we want to do in the next years. This is a real good question. Not for tomorrow. Okay.
So thank you, Jesus. So we now have finished the first round of our event, and we'll have a break for lunch. So now I think we can come back here at 3:15. It would be like 90 minutes.
Who's the first one to present? No, Paulo is the last one.
The next one is Mariana Vidal, and then Paulo Guimarães Jr., and then Clementina Gavetska.
Okay, we just need to tell Paulo
Perfect. So then 3:15. Is that okay? Good. Thank you.
Sí, seguro, seguro. ¿Y puedo quitar yo? No, tú me dices. Tem alguns minutos, ele vai se preparando.
Hello, welcome back everyone.
My name is Ana Paula, for those who not know me yet. And I am also a researcher at the Towards Dilution Landscape Project. And I will be representing the other speakers from this hour to the end of the symposium. And first, I would like to introduce you to Mariana Vidal. Dr. Mariana Vidal holds a PhD in ecology from the Institute of Bioscience at the University of São Paulo, with a doctoral stay at the Center for Research on Desertification of the Valencia University in Spain, and a postdoc at the Oswaldo Cruz Institute from the Oswaldo Cruz Foundation. In Brazil, she is interested in the possible effects of habitat loss and fragmentation on interaction systems. Thank you, Mariana. I will just let you do. Here. Control F.
Hello, thank you very much for the invitation. Thank you very much for the invitation. I'm really happy to be here. And today I'm going to talk about my main interests in research. And my main results encompassing the effects of landscape structure on ecological networks. So when we think about biotic interactions, we recognize that they all occur locally. However, it's too loud. Or is it too loud? No? What? Thank you. So interactions occur locally. However, the individual behavior, the population biology, and community dynamics of those interacting organisms are often affected by the spatial distribution of resources at a broader landscape scale. So the landscape structure, that is the arrangement of land uses and land cover across the landscape, it affects the habitat quality, the relationships between organisms and their habitats, and patterns of species co-occurrence. So landscape structure may deeply affect the outcomes and strength of species interactions. And the processes of habitat loss and fragmentation may cause important changes in landscape structure. So we can move from a pristine area covered entirely by native vegetation to fragmented landscapes with varying degrees of fragmentation. And all these changes in landscape structure may cause changes in abundance, extinctions, and changes in behavior, all of them with important consequences to biotic interactions. So here's an example of how habitat loss can affect seed dispersal systems. Here we have a gradient of patch size. So in this case we have large fragment moving to smaller fragments, and we can see that the birds, the frugivorous birds, respond to this change in habitat cover. So in this scenario, the large frugivores here are super generalist species. They are absent for small patches. And these have consequences to their interactions and the interactions that we observe in these patches. So the organization of such interactions change in this gradient. So we can observe reduction in degree as we lose forest here, and maybe increasing modularity, and reduction in nestedness as we lose these super generalists. And I'll illustrate the effect of distance from forest here in a system of bee communities visiting coffee plants. So there is empirical evidence of the effects of the diversity of pollinating bees and an important increase in coffee fruit set. And bee diversity is often associated with forest cover, as forests are sources of nesting sites and floral resources. So it has been demonstrated also that bee diversity declines with distance from forest. And here we were interested in finding a critical distance where the bee community would change dramatically. So we sampled bees in coffee plantations at varying distance from forests. And according to our expectations, what we saw was this decline in bee species richness as we move away from the forest. And this pattern was consistent among 4 years of sampling. And we built this matrix of bee species across this gradient of distance. And as expected, Apis mellifera was found throughout this distance gradient. And looking for this critical distance, we analyzed the nestedness of this matrix. So we analyzed nestedness among columns of this matrix, and we gradually removed the most distant points, and we kept analyzing this nestedness. And what we found that was a significant nestedness until we reached a distance of about 43 meters from the forest. Then this nestedness lost significance, indicating that our new model was able to reproduce the observed pattern. So up to 50 meters away from the forest, the composition of bee species is relatively homogeneous. As we enter the coffee plantations and move away from the forest, we observe impoverished communities with only subsets of those species that we see closer to the forest. So it's a really small range where we can find homogeneous communities. And another example of how landscape may affect the interactions are in this paper, where we analyzed how habitat loss changed plant frugivore networks. And so here we wanted to understand how extinctions triggered by habitat loss reorganize species rich interacting assemblages of plants and frugivorous birds. So we estimated the realistic sequence of bird species extinctions following habitat loss, and we simulated this sequence of extinctions in a pristine network, a pristine frugivory network. We had several sample points and calculated the percentage of forest cover around each sample point. And according to the minimum percentage of forest cover of each species, where each species occurred, we estimated this sequence of species extinctions. And what we found was these threshold responses. So the number of species, number of interactions, connectance, and nestedness all showed this threshold response to the proportion of habitat cover. And they all occurred around 30 or 40% of forests. So when we reached this amount of forest in the landscape, the structure, the organization of the interactions changed. And our new model that are here in the box plots clearly reproduced the same patterns. So here, the number of species expected to go extinct following habitat loss, rather than specific order of extinction, determines the structural impacts over frugivory systems. And we found the threshold of first cover for the organization of frugivorous systems that is always about 30 to 40% of first cover. And this indicates a need to maintain first cover over this amount if we are to conserve the organization of the interactions. We also evaluated the robustness of our network. Robustness is the system's tolerance to species extinctions, and what we usually do is to build this curve that is plotting the percentage of surviving plants in function of the percentage of primary bird extinctions, and we calculate the area below this curve, and areas that are close to 1 indicate that networks are relatively robust to primary extinctions, because we have to lose many species from in this case the birds, to lose some significant species, plant species. And areas close to 0 indicates very fragile communities, because if we lose few bird species, we cause large extinctions, important extinctions in the other group. So here our realistic sequence of extinctions was between these extremes. No? Here we have removal from most to least linked species, that is the most critical, the worst scenario, and here we have the removal from the least to the most linked species. So our realistic sequence was in between these two. And the last project that I'm currently working on is changes in the host parasite meta-web due to habitat loss. So here we wanted to understand how small mammal communities respond to first loss, and how their networks with parasites also change with first loss, and what are the implications for spillover risk. So we worked with more than 200 landscapes with small mammal species data, and we calculated the percentage of first cover in each one of these landscapes. And what we found, here we have on the y-axis the small mammal species, and here a gradient of first cover from 100% to 0, and we classified these species as forest specialists, those that occurred above a certain critical value of habitat, and the others as disturbance adapted species. And we used this distinction to simulate the change in our mammal community. So here we have this we have this gradient of first cover from 100% to 10% at most, and what we did was, as we move from this scenario to the first state landscapes, the host communities, the small mammal communities, will change. So for the first specialists, what happened is a sequence of extinction, similarly to the other approach. So as we lose first, some species are lost from the system, but also enter the others enter the systems, those disturbance adapted. According to their maximum percentage of first cover, we added them to the system as we lose first. And we analyzed how their interactions with parasites changes in this gradient. We used a recently recently published database of host parasite published by Cecilia's group, and we simulated this sequence and this sequence of extinctions and introductions of species in this meta-web. And what we found was that for the small mammal species, we saw a peak of richness at around 60% of first cover, and then the species richness started to decline. And for the parasite species, the peak was around 10% of first cover. So despite the fewer hosts, we observed growing numbers of parasites in these deforested landscapes. The number of interactions presented a similar pattern. The connectors increased a lot at 26% of first cover, indicating that maybe first loss may speed up parasite transmission rates. And in nestedness, we saw this increase also here at 38% of first cover. And this increased nestedness may indicate a concentration of interactions by the most connected species as we lose habitat. And modularity didn't show a significant pattern. When we look at small mammal species that occurred throughout the entire gradient, we see that their closeness centrality, it increased a lot as we lose forest. So maybe deforestation may accelerate the infection spreading among potential hosts. Here it's the same pattern, but when we look at all those host species, some occurred at the beginning of the gradient, others at the end, but we can see that there is the same trend to increase the species closeness, the host species closeness. So, despite important decrease in small mammal richness when first cover is reduced to around 60%, the number of parasites kept growing until forest was reduced to 10%. I think it's really interesting because we have fewer hosts but more parasites. And first loss seems to benefit species with many interactions. And first loss may speed up parasite transmission rates and accelerate infection spreading among potential hosts. And the next steps here that we are working on is that our results point to this trend that first loss leads to decreased biodiversity, but an increase in zoonotic spillover risk. And we expect that with forest restoration, maybe we increase biodiversity and this decrease the zoonotic spillover risk. But we know that this response of disease risk to time, and since the beginning of the restoration process, we can observe different responses. So maybe the disease risk may even increase in the beginning of the process. That would be this line, this yellow line. Maybe because we have some new species entering the system, what may cause an increase in the disease risk. But as first starts to get older and more structured, maybe this risk may decrease. So we have many different possibilities, and with data, with empirical data, we are trying to answer these questions. So thank you all, and
does anybody have questions? No, it's here with me. Very nice talk, Mari. Thank you very much. I don't know if I understood correctly, so that's why I'm making my first question first, and then I make the second one if it makes sense. So, different of what happened with the frugivory networks, what I understood is that in the host parasite networks, the species that remain in a very fragmented landscape are those that are more generalist, is it?
Can Can you hear me? Yes.
Yeah.
And in the frugivorous networks, we only looked at those forest specialist species. So we expected that the species only go extinct in this gradient of habitat loss. And here in the host parasite network, what we did was to remove species, those forest specialists, and also we added species that weren't present in the pristine area where we had continuous forests. So we started with a community with only forest specialists and those disturbance adapted species that occurred in 100% habitat cover, and beginning from this scenario we started to remove and to add species.
So when you add these species that are not on the pristine forest, these species are generalists for some reason, I guess.
Are generalists of habitat. No, they are not generalists in the network. Yeah, they are habitat generalists, those species that are entering the system. Because as we lose forest, what we expect to see is these species that can tolerate other land covers will enter the system. So those that use agricultural areas or urban areas will start to enter this host parasite network, and these species were absent when we had only forest.
But then in the end, they are these species that enter, they make the environment available for the parasites to still remain this deforested area, is it?
Yes.
Okay.
What we see is that they are species with many parasites, so they enter the system, so the number of parasites keep growing, even if we lose a potential host species.
And another question that I have, oh my god, sorry guys, it's just the last one. I didn't understand, so when you did the simulation, did you simulate the infection or the species loss in the network? Do you simulate like new infections?
No. No, we had this meta web of interactions that are all the interactions that were recorded in the literature. So this species usually, this species was ever recorded with these and these parasites. So we used this network with potential interactions, and we simulated only the changes in the host community. So the hosts were added or deleted from the system, and we observed how the network changed with these deletions and addings.
Sean, thank you for the talk. If I might, you've shown that. Okay.
It's the cell phone.
Use the phone. Okay. You've shown that there is some, both with the bees and with the other samples, there seem to be some thresholds, and right now there's a hot debate with the fragmentation to try to disentangle if. Sorry. So I can escape the speaker. Okay, I can come here. Sorry. I hope I don't lose my mobile. So there is a strong debate now within the fragmentation in conservation biology that has to do if the effect has to do with habitat loss or has to do with fragmentation per se. And I don't know if you have data. I mean, you have quite a lot of landscapes that you can look to see how much of the effect has to do with habitat loss or how much has to do with fragmentation, because it seems, I don't know, that this threshold might be the point where actually it's not habitat loss but fragmentations when the effects start. Do you have any idea? Thank you.
Yes, we in the field,
habitat loss is very often accompanied by habitat fragmentation. So it's difficult to disentangle these. But here we used only percentage of forest cover in these landscapes. But another thing that we are still working on is to look at the edge amount that reflects somehow the fragmentation process. The amount of edges that we found in these landscapes. So we made some exploratory analysis, and it's the pattern in the structure of the community isn't very clear when we try to use instead of the gradient of forest cover, we use the quantity of density of edges in the landscape. The pattern isn't very clear, but to simulate these changes, but we have to work more on this to incorporate the fragmentation process, because we know that for these species, these contact areas are really important for them to occur. Maybe we know that some species prefer areas that have these edge effects with these edge areas between forest and field, agricultural fields, and I think it's important to explore this better. Here it's just forest cover, but I'm sure there is some effects of fragmentation also. Thank you all.
Acho que a gente que tem que fazer.
Não, a gente, que ele não vê a gente. Okay.
Paulo, can you hear me?
Yes.
Yes? Okay. Thank you.
I don't know, put him in full screen.
Okay, I open Paulo. Okay. Thank you, Mariana, for your great talk. And for the following talk of our event, I would like to introduce you to Dr. Paulo Guimarães Jr., who will give his talk online. He's here now with us. And Paulo Guimarães is a full professor at the Ecology Department at the Institute of Bioscience at the University of São Paulo, Brazil. His research interests involve integrating data in the natural history of ecological interactions, network science, and mathematical modeling to understand how ecological networks shape and are shaped by ecological and coevolutionary processes. Paulo, when you are ready. I think if you have something to share, you can. Yeah, great. Let me see. No, maybe here.
Now,
yeah. Now it's better?
So now he's muted.
No, he's yeah.
Are you guys listening?
Yes, we just. How can we increase here?
Maybe it has to be in the present mode.
Yeah.
Pantalla completa.
Não sei.
Foco.
Yeah.
Foco. Wait, yeah.
Here? It's better here? Yeah? Okay, you can begin anytime, Paulo.
Okay, thank you guys. Well, first of all, thank you, Anna, for the introduction. I would like to be I would love to be in Madrid right now. I was a problem of flying schedules, to be honest. But I'm glad I'm able to participate even through Zoom presentations. I would like to thank Cecilia for the invitation. Today I would like to talk about indirect effects in ecological networks. But to do so, I would like to invite you guys to fly to California, and there is a beautiful submarine forest, the kelp forest. The kelp forest is one of the best examples of systems in which indirect effects are known to play a fundamental role. The astonishing diversity of organisms in these forests contrasts to virtually a desert in similar environmental conditions. These two alternative states of the system, of the same system, is governed, at least in the North Pacific coast, by the abundance of herbivores, especially sea urchins. But it's not actually the abundances of sea urchins that shape the form of this and the organization of these ecosystems. It's an ecological interaction. It's actually a set of ecological interactions that are organized in a chain, leading to indirect effects. And one of the main players is this beautiful mammal, the sea otter. Sea otter prey upon urchins, and this makes the completely difference to which stage you observe in nature. If you observe the astonishing diverse kelp forest or a barren seafloor that's essentially a desert with the presence of high abundance of sea urchins. This is the work of the life of my good friend Jim Estes, and once I asked him to draw what he knows about the kelp forest system in a single cartoon. So this is more than 40 years of studies with several players, and actually I framed this to remind me the complexity of food web. So it's in my office at USP. These three nodes here, marked with the red rectangle, the otters, the urchins, and the kelp, are the key elements of the system. So this leads to the define indirect effects. So indirect effects are a chain of effects that propagates through the network, like sea otters control urchins that control kelp forest. So the negative effect of sea otter on urchins leads to a positive effect on the kelp forest. So I would argue that this is the main consequence to be part of a network. Is to the fate of a given element of this network is not dependent on its own properties alone. It's not only a consequence of the pairwise interactions, but it also depends on other elements of the system that do not interact directly with the focal element, like the fate of kelps and the fish that rely upon kelp forests rely upon the presences of sea otters. And parasites are one of the most fantastic examples of how these indirect effects can occur, because many parasites have complex lifestyles that involve several species. So they are actually surfing in a network, moving from node to node. And because you have multiple parasites and multiple hosts in a single place, and those parasites can use multiple vectors as well, you create a network in which these indirect effects can propagate. Moreover, parasites are embedded not only in ecological networks, but also in social networks and spatial networks. And so this creates the possibility of effects propagating to different types of networks. But if the effects are propagating to networks, at some point the structure of those networks may play a fundamental role. This is the most fundamental assumption of the network science, as encapsulated by this quotation by Kaka Albert and Albert Bardazzi. So if a network display some organizing principles. So the dynamics and the structure of this network these principles will be at some point encoded using some routing topology. So today we'll focus on two aspects of indirect effects in ecological networks. I first focus on how indirect pathways that allow these effects to propagate are related to fundamental architectural laws that apply to any kind of network. And then I will talk about the two types of indirect effects that you can coarsely divide these effects, and you can study how these effects propagate in the system. So, the first thing that I would like to point out is that indirect effects are propagating across network pathways. And those network pathways are actually emergent properties of architectural laws. So, here I use some examples that are local networks of particular type of species interactions. So if you look in nature, those networks come in two different flavors. Or they are a set of disconnected small group of species interacting, like the plant involved insects or the mimicophytes and interactions. Or they form a network in which there are indirect pathways connecting all elements of the system, as we observe in predator-prey interactions, in seed-dispersal systems, and also in some parasite host networks. These two flavors are actually two states of networks, and they show up over and over again in different types of systems. One state is called the disconnected network, is characterized by the presence of multiple components, these groups of isolated nodes here. And the second one is characterized by the presence of a giant component. And the network is called connected network. And I would say those two types of network are phases. It's not a metaphor. It's actually an example of phase transition as we observe between ice and water, or between iron and a magneto. Okay, it's a well-known example of phase transition. There is an entire theory that we networks to describe the transition between these two states. So here I will plot this transition in a simplified way. So in the x-axis we have the connectivity parameter, and 0 is the critical point predicted by the theory of random networks. Negative values implies disconnected networks, and these negative values are associated with this parameter to low variance in the number of interactions per species. Positive values imply high variance in the number of interactions per species. When we plot empirical networks, empirical ecological networks, you observe that these empirical networks follow the prediction. So the black nodes here represent networks that theory predicts disconnected networks. So those are negative values for the connectivity parameter. So once you cross 0, there is a lot of variance in 0 because you are in the critical point. When you increase, these values get more and more positive. All networks become characterized by a giant component. So the red nodes are networks that are predicted to be characterized by a giant component, and you can see as positive as the connectivity parameter, more sure you get that you end up with a network with pathways connecting directly or indirectly species in the system. So the network, the green network, the network with positive values, is the network where indirect effects can propagate. So what is happening here? So what we hypothesize is that natural selection in some systems are constraining the number of partners to not allow generalization. So as in some kinds of plant herbivore interactions, especially some plant insect interactions, and also interactions that involve ants and plants. In this case, you end up with a network that is disconnected. But if selection sometimes fails in highly corrupted species, like frugivores that feed on multiple fruit species, or predators that take down multiple types of prey. Some species, not many, but at least some species are present, then you have a connected network. And these highly connected species, therefore, are building up a network in which they use indirect pathways. So the most famous example of those highly connected species are the keystone species, like the sea otter that feed upon a huge diversity of invertebrates, the sea urchin, and the wolves. So these highly connected species are actually creating the network, a network in which effects can propagate. But one interesting thing about highly connected species is that they not only create direct pathways, they create indirect pathways. So this is a study of C-dispersal networks led by the lab of Alexandre Pires in Brazil, in which they study the consequences of the reintroduction of some frugivores that die out in one forest fragment in Rio de Janeiro, to be accurate. And in the plot, the number in the x-axis indicates the number of pathways with length 2, 3, or 4, 5 that are created by those species. So once you introduce a toucan, you create a lot of direct interactions, but you create much more indirect interactions that involve the toucan and a plant and another frugivore. This will be interaction of length 2. And with length 3, you involve a second plant species, and so on. So you are building up, you are creating integration in the network. So there is ways of estimating these indirect pathways and relate that to structure. For example, you can see that the increase of number of pathways connecting species, like the long pathways connecting species in the network, is a consequence of the structure of this network and can be captured by the leading eigenvalue of the matrix that describe the patterns of interaction. So we call this pathway proliferation because there are much more indirect pathways and direct pathways in the system, and there are even more pathways if there are long pathways involving multiple direct interactions. So as the leading eigenvalue increase of this matrix of interactions, increase as well the likelihood of the network to be connected, and also the number of pathways. Parasites. So we know that many parasites are thought to be the ultimate example of specialization, but we also know that some highly specialized parasites that with physiological sense use multiple hosts. So the relationship between specialization and the number of hosts is not one-to-one. Also, we know that in parasite host networks, as Cecilia showed before, there are highly connected hosts and parasites. And even more interesting that some parasites connect different trophic levels, adding up pathways across this network, both direct and indirect pathways. We have no clue about the consequences of the way parasites create these pathways in the system, but they probably will increase the importance of indirect effects, the effects that propagates in these pathways. But how can we estimate these indirect effects? So, first of all, there is two coarse classes of indirect effects. And before I start with that part, this last part of the talk, I'd like to thank and to make clear that this is not my work, this is the work of dozens of researchers across the world that have been helping me out and teaching me how to explore those networks in a myriad of problems, mostly with co-evolution, but also conservation biology, restoration ecology, and disease spread. So keep in mind that when I say we, I'm saying there. Okay. So the first type of indirect effects is the effect that is caused by the extinction of sea otter. We can call this a snowball effect. So here, what happened in Alaska, at least decades of evidence suggests. The raining industry led to the die-off of several whale species. This changed the diet of killer whales that start to feed upon sea otters. Then sea otters, who is a small component in terms of biomass of those kelp forests, die out. This should increase in the abundance of urchins, much more increase in the biomass of urchins than the loss of biomass of sea otters. And then this leads to a completely knockdown of the kelp forests, the fish stocks, and everything around those forests. Even more bigger change in biomass. So this is an effect that is increasing through the pathways. And this is really hard to predict. This is really hard to understand. And we are just beginning to explore this type of problem. A second type of indirect effect works like the telephone game, in which a person is telling a message, and there is a probability that the message is changing through that chain of people. And usually, if the length is long enough, or if the people are nasty enough, this message will be changed pretty fast. So in this case, the effect dies off through the length of the pathway. It's like energy flowing to food webs, but the energy is being lost in terms of heat in the maintenance of biological systems as you move to different trophic levels. So in this case, the effect is not amplified as in this mode, the effect is actually decaying. And this is easier to work on it. So starting with the observation that ecological networks, as any type of networks, show pathway proliferation, we can then add in the assumption that the effects decay along the pathway to explore the consequences of indirect effects. So this assumption, imagine that we are modeling the risk of extinction of a species in a network, like a mutualistic network, so plants being dispersed by frugivores. So the effect of the loss of the plant on the frugivores will be big. There is a probability that these frugivores die out. But then there is a probability that another plant species die out because the seed dispersers die out before. But this is a multiplication of probabilities, and by definition, this multiplication of probabilities will be a number that will be equal or smaller than the risk of the seed dispersers die out. And we can go on, and this effect is just dying off with the length of the pathway. So if you assume that, you can mathematically compute a matrix that describes the sum of the direct and indirect effects. This matrix is related to the matrix of direct effects, but it is not a simple relationship. So here, Q is the matrix of direct effects. So the species are in the rows and in the columns. The hotter the column, the stronger is interaction. Then, if you compute the inverse of the identity matrix means this matrix, you get actually the matrix that describes all pathways from direct pathways to the infinity connecting species. And as you can see, most of the species are connected by weak in brief interactions, but the matrix is completely integrated. So you can use this matrix to try to understand the consequences of indirect pathways connecting species in the network. And this is not a real result. I mean, we got this result in a co-evolutionary model, but then in the 90s, a group of Japanese researchers got that described a similar result in the same matrix, the very same matrix, describing energy flow in food labs. And before, Vasily Vorontsiev won a Nobel Prize because he used this matrix to develop the input-output analysis in economics to understand the flow of supplies and consumption in economic systems. And even before, Katz created a measure of centrality based upon the same matrix that we describe the social influence of a person. And this is the root of the algorithms that today we use to search information in the internet. In our lab, we have been using this approach to several problems, from co-evolution to co-extinction dynamics, and also some work with disease spread. And some people in my lab, and former students as well, have used that during the COVID pandemics to understand the risk of propagation of COVID to different regions, to the social and spatial networks connecting cities in Brazil. Here, just an example of what you can do with this approach. This is the result of a co-evolutionary model, so I'm interested in understand how the network shape the trait evolution of species, in this case are mutualistic species, so they have traits in the model, and those traits are just numbers, but the network are empirical networks. So, and then I can ask the question, when indirect effects network matter? But I do not know in nature when indirect effects matter to co-evolution. I cannot do the experiment or observe the change in a cat forest, but I can explore when, in which scenarios indirect effects matter. So I can, for example, assume that the selection posed by mutualism is very weak. And then we can increase the selection and see what is the contribution of indirect effects and direct effects. So here, each point is a average of simulations parameterized with a given ecological network, empirical network, mutualistic network. X-axis describes the increase of selection posed by mutualistic direct interactions. And y-axis measures how much of the trait change in the simulation is a result of direct or indirect effects. And as you can see, as you increase the importance of mutualistic interactions as a source of selection, you also increase the importance of indirect effects. So I do not know if indirect effects are shaping the co-evolution of mutualisms, but I'm pretty sure to assume that at least for a wide variety of parameters and model types, it's really difficult to find a situation in which selection is imposed by mutualisms is strong and indirect effects are not. Or the opposite, to be honest. Most of the time, it's the selection imposed by mutualistic interaction, the direct selection imposed by them are strong, then much of trait evolution is actually a result of indirect effects. And again, parasites. So, first of all, we are now starting to try to generalize the telephone game model or approach for parasite-host interaction. There is some change that we need to do in the model, the underlying evolutionary model that we are using, but apparently we can do that. Secondly, much more complicated is how can we tackle snowball effects, those effects that we need to use change in the system. Those probably are the most important effects in terms of substantially change our ecosystem. And I think that parasites may be key to understand how indirect effects are propagating and connecting different types of networks, especially spatial, social, and ecological networks. But again, we do not know the potential consequences of incorporating parasites to those dynamics. I'd like to finish thanking the network of collaborations that make this work possible. My former supervisors and my collaborators, many of them, Flávia, Cecília, Sabrina, Mari, Ana as well, are there with you guys. The lab, the prep, the equipment, and the former students, and the family agencies and institutions that make our work possible. Thank you so much. I'm very glad to take answer questions if there is time.
Do we have any questions? Yeah, I think I moved, but do we have any questions? Yeah, I tried to. It's better from there. Yeah. Yeah. Then he can see. Okay.
Hi, Paulo.
Yes, everyone.
So, okay. Okay, I see you there. Okay, before you, you have Mariana talk about the parasite in a network that change through landscapes. And so new association occur in different scenarios of fragmentation. So when you do your indirect effect, are you considering this changing of possibility of change of interaction?
Yeah, well, yes, in two ways. Like first, we can, if we assume a evolutionary or ecological underlying model, the model can allow interaction wiring and or change the interaction strength. If there is a steady state, the system approach to that, then we can analyze at the equilibrium and make predictions based upon compute the indirect effects in the equilibrium of the simulation. Also, we can assume the network, even if the network has no steady state, if the network is changing, we can try to measure the current matrix and make predictions in a short term. Interesting, this apparently works pretty well in some set of models. For example, when we use the same approach for co-extinction cascades, so have in this case mutualistic partners, when species die out, then this leads to a co-extinction of a partner with some probability. So when you start the co-extinction cascade, the matrix is changing because you are literally removing species and links, but the predictions based upon the original matrix allow us to predict which are the species that are more likely to go extinct if one particular species is removed from the network, even with the network changing. So I would say that the approach assumes that you end up with a fixed matrix. They don't need to be fixed from the beginning. But even if the matrix is not fixed, there is some intuition that you can get from the dynamics. About the dynamics, sorry.
Thank you.
Eu que agradeço.
Oops, oi miúdo.
Oi, Flávia.
So I don't know where I look at you. Where is the camera? It's here.
I'm everywhere.
So yeah, you are everywhere. There are 3 screens here. My question is regarding the last part when you said about the host-parasite interactions. How is the snowball effect? I don't know if you call this snowball effect, but snowball effect on the host parasites. I think I understood this, and I understand your explanation when you are telling us the story about the kelps and the sea otters and the whales and everything, like a trophic interaction. But when you go to the host parasite, for me at least, sounds like many balls and not exactly a snowball. Can you make an argument about how this could be?
Yeah. I think I was not clear. I was trying to say that I do believe that both types of dynamics can be observed in any kind of network. So I would argue that the telephone game is probably the most common type of effect, because it's just assuming that a change, like measuring abundances and prevalence of parasites being affected, and the parasite of the parasite or the prey of the host. There is some propagation, but maybe this propagation is dying off with the length, because there is some kind of self loop or some kind of negative feedback that keeps the abundance controlled, then we should expect those effects die off. But when I'm talking about the snowball effects, is that they may occur, and when they occur, they change everything. So imagine, there is one example, it's not a parasite, but it was just out in Science, I think, last week. that sounds that we can push a little bit and treat that as a parasite. So there is an invasive ant species that from Mauritius that invaded Africa. And it's taking over the acacias. Those acacias host ants that protect them against enemies. But those invasive ants doesn't do anything. It just exploit the system as a parasite. So they go there and take over the acacia. Okay? So this is a very small change. But probably the biomass of ants are not changing much. They're just changing from one species to the other. But then what happens? Elephants can go there and now can feed upon acacia. They just destroy the entire plant. So the physiognomy is changing completely. So you change the diet of the elephant. You increase the biomass is removing, but then you change completely the environment. The environment now is open. And now this effect is changing behavior as well. It's another aspect. What? Lions are not being able to kill zebras anymore because it is in an open landscape, so they shift the diet to other prey, to buffaloes. So the effect is that this completely crazy dynamics that at least from ants to plants to elephants is apparently is an increase in the effect. I can imagine that something similar could happen with a parasite, a more traditional parasite.
Oh yeah, okay. And my is there any other question? I'm making a lot of questions. When you were talking about the indirect effect and direct effect, I was thinking about some interactions of parasites and hosts. They actually happen because the parasitism happens because the predator preys a prey that has a parasite, and the cycle completes in the host, for instance, in the predator. Sorry. So, do you think this is a kind of indirect effect, or for me it's very direct? But it's direct, and also I don't know if you can call this like a high order interaction. It's something that depends on the interaction that is happening between host and parasite in the first time, the host, the prey host. Do you think this kind of effect can be calculated with the indirect or with the direct effects of those matrices that you showed?
I think so. I agree with you. There are actually multiple ways of describing it. I would argue that, at least for me, the most intuitive way will be describe the parasites as something that is flowing through a network. But this is a very biased kind of view from my perspective, because I'm used to the interactions between the big things in the environment, so I see parasites just flowing there and change the properties. And then we can describe that as a network flow. The network flow is strongly related to this approach. Like the matrix I show at the end, the inverse of the identity matrix means the matrix of interactions, is actually pretty much similar to the Laplacian matrix that describes the flow of something in a network, like information, a fluid, or anything in a network. So first, I think the approaches are related in the sense that the network diffusion is not so far away from the approach of direct indirect effects I show. And second, you can actually build up, generalize this approach to networks that have different types of interactions. So you can describe the parasite interactions in one way, and then the trophic interactions in another way. And then the key is how you move through these different types of links. And I think this is a big challenge for any of those kind of multi-layer kind of approaches, because the big difficulty is how is the coherence ratio between something that is flowing in one network to something that's flowing in another network. In my lab, we actually had some multi-layer networks that we studied, especially by Ana Paula Cis and Lucas Medeiros. Ana Paula studied coevolution in a multi-trait kind of model, and Lucas in a single trait, but across the space. So we have, in both cases, we have two types of interactions. The species interactions, and then in the case of Ana Paula, the genetic correlations. In the case of Lucas, the migration among individuals. But because in our models we can think and describe both processes as changing traits, we are working with the same currency.
Okay.
But in principle, I would think that we could use this approach. And the trick would be which kind of description will provide more insight. Because those networks are just abstractions anyway, isn't it? They are not in there. They are not like networks of types. They are just metaphors of patterns of interaction among individuals that we observe in nature. So which description will be more informative? And I don't know which one.
Okay, these were my questions. Thank you, Paulo, for this great talk. And now I will not show you anymore.
Okay, thank you guys.
But you can continue with us.
Thank you so much. Thank you everyone for the invitation and also for making it possible to give a talk in a remote way. I'm a bit emotional as I was, because I saw so many people that I like giving talks, that good former students, good friends, and colleagues from live colleagues like Sabrina is a former live colleague. So it was pretty special to participate, even if in a remote way. Thank you guys.
Thank you. And now for our last talk, I will like to introduce you to Klementina Gavetska. Klementina holds a PhD in numerical modeling of geothermal energy systems at Imperial College London, and currently she is a postdoctoral researcher at the Bascom Lab at the University of Zurich. Her research combines numerical modeling with an interest in ecology and conservation by developing meta-community models to study the consequence of habitat fragmentation and its subsequent restoration on ecological networks and ecosystem functioning. Thank you, Claire. We have desktop somewhere. Oh, thank you. Okay, thank you. Thanks.
Well, firstly, thank you very much for this invitation to this workshop. It's been a privilege to be part of it and learn from you, and now be able to share a bit of my work with you. Well, I prepared a whole slide to introduce myself, but I guess Ana Paula has done it beautifully. But yes, so as she explained, I'm actually not a classically trained ecologist. I began by studying civil and environmental engineering, and I also did my PhD in this field. But what I did throughout this time was to essentially use lab and field data, so empirical data, to build models which describe engineering problems, and then use these model simulations to guide engineering design. And then at some point I became fascinated with ecology and communities, ecological communities, and the complexity of interactions within them. So I decided to do a master's in environmental science at the University of Zurich, and now I work as a postdoc with Jordi Bascomte. And what I try to do is kind of follow the similar procedure. So develop models that describe how communities change under global change, and I focus on habitat transformation. And my aim is kind of to use these models to guide provide some like practical recommendations for conservation. And the framework that I have been working with is metapopulation dynamics. So this framework assumes that species inhabit a fragmented heterogeneous landscape, and at any given time, some patches in these landscapes might be occupied by a certain species. but some patches might be empty. So this is a patch occupancy metapopulation framework. But as individuals move or disperse across these landscapes, the empty patches can become colonized at the next time step, so for example like this one here, and then other patches might become empty due to some local extinction events. So understanding this balance between the extinction and colonization dynamics is really at the core of understanding the persistence of metapopulations in fragmented landscapes. And the first model that kind of tried to tackle this, at least in ecology, was developed by Richard Levins and then later modified by others. But essentially it describes the rate of change of the fraction of occupied patches in a landscape. And this rate of change is essentially a balance between the colonization. So this is the first, oh wait, you can't see the, oh here. So this is the first term, colonizations, which is the positive term in this equation, and the second term, the extinctions. So it's the balance between colonization and extinctions. And then C and E are the colonization and extinction rates, respectively. And this term in the brackets here is essentially tells us the amount of habitat that can be colonized. So these are patches that are empty, but also non-destroyed, so pristine habitats. So we can now use this model to, if we vary D, so the fraction of destroyed patches, we can study the effects of habitat loss on the persistence of a metapopulation. And this model showed that as habitat is lost, so the amount of habitat reduces, at some point there's this critical threshold at which the metapopulation becomes extinct. And this is what we call the extinction threshold. And what it essentially means that for a metapopulation to persist, we need some minimum amount of habitat. And this depends on again, on this balance between extinctions and colonizations. And this is actually equivalent to the epidemiological threshold that was mentioned before. But this is basically a very nice example that, well, I like this example because it shows how a very, very simple model can provide us some insights into a very practical problem, which is habitat loss, and then provide recommendations on habitat protection. Now, this model was what we call spatially implicit. So it assumes that there's an infinite number of patches and that species can colonize any patch in the landscape. But of course, in real life, landscapes are not infinite, and also perhaps patches that are close to each other are more likely to become, or dispersal is more likely to happen between patches that are close to each other. So this next model incorporates space into the metapopulation framework. So here we have landscapes with, starting with a pristine landscape where all patches are pristine. And in this study, this landscape was destroyed in steps until almost the whole landscape was destroyed. And what the study very nicely showed was that there's a sudden point during this process at which the landscape fragments. So this pristine habitat starts to break up into many small clusters of patches. And when we simulate the extinction colonization dynamics on this landscape, what they found was that the extinction threshold was much lower than that predicted by the spatially implicit model. So the model that didn't take space into account. So essentially, we would be underestimating overestimating the extinction threshold if we don't take into account the connectivity of habitat patches. So it also showed that, it also showed that basically it's not just the habitat amount, but also the arrangement of habitat patches in space that determine the persistence of metapopulation. Now, of course, species don't exist in isolation, so they exist in communities where species depend on each other. And this can be very important for their dispersal dynamics across the landscape. So one species might depend on another species' presence for dispersal and colonization of new patches. And we can incorporate that into our models using network theory. So now we can model these interactions on both the local scale, also the meta-community scale, by modeling local and meta-communities as networks, so where the links are integrated and interaction between species. And going back to this habitat destruction problem, that's what this study did. So it incorporated these interaction networks. Again, it simulated the process of habitat destruction. And by that, what that allowed was them to study the collapse of the entire communities as the habitat was destroyed. And for example, one finding that they found was that this mutualistic community, the extinction threshold, increased with metacommunity size and also network connectance. So larger networks that had more interactions were more robust to destruction of habitat. And what I'm very interested in is also habitat restoration. So we're currently in the UN decade on ecosystem restoration. So there's a big push to restore our natural environments. And what we wanted to do was to kind of try to understand how communities might recover in these newly restored landscapes, and then provide some kind of insights into how we can improve restoration strategies. So again, we took the same model, but now instead of just destroying it, after destroying it, we started to restore this landscape. And we simulated this with two different types of networks. So communities, one was a mutualistic community, one was antagonistic on the other side. And what I'm plotting here is on the vertical axis, the fraction of occupied patches. So the gray line, which you can barely see here, shows how this fraction of occupied patches reduces as we destroy the habitat. And the blue line shows how it then increases if we start restoring the habitat from here to here. And what we found is that in the case of mutualistic communities, we saw that at the initial stages of restoration, the abundance or the fraction of occupied patches was lower than it was when the habitat was being destroyed. And so we call this restoration lag. But on the other hand, in antagonistic systems, what we found that the fraction of occupied patches was actually higher than when habitat was being destroyed. So we had this boost in abundance of species. And this actually happened because it's an antagonistic system. So as we were destroying the habitat, the abundance of consumers reduced. Even some consumers went extinct, which means that during restoration, the resources could bounce back, so they had less pressure, so they could recover much more easily. And so what we also found was that this lag or this boost, the magnitude of it, depended on three things. So firstly, the amount of destroyed habitat. So the more destroyed the habitat was when we started restoration, the larger this lag or boost was. Secondly, the meta-community type and structure. So for example, in mutualistic communities, we found that larger, more connected and monested communities has smaller lags, so they recovered more easily than smaller communities and more modular communities. And finally, the spatial restoration strategy. So the way, the order in which we restore patches. And we found that it was much more beneficial to restore patches in clusters rather than creating kind of a more fragmented matrix, which kind of makes sense, I think. Now, so far we only kind of modeled ecological dynamics, but we know that both ecology and evolution affect each other, creating these feedbacks, these loops. So for example, thinking of this meta-community framework, we know that ecological processes can affect the local species composition and structure. And since species co-evolve inside communities, any changes to community structure will affect the co-evolutionary trajectories and therefore any changes in species traits. And these traits in turn might affect how these species disperse across the landscape. So it could affect the colonization and extinction dynamics, which could then in turn affect the ecological processes, so any changes in abundance. So we wanted to incorporate this feedback into our models, and we simulated the ecological extinction colonization dynamics just as before. But now we added another step where the species were allowed to co-evolve inside each patch, so with the local community. And the way we modeled it was using the models developed by Paolo and Cecilia. So this was the model that was mentioned a few times today already. But essentially what it assumes is that each population, a local population, has a mean trait value, Z, and that evolves over time. And that it evolves due to two different selection pressures. So we have the selection due to species interactions, and on the other hand, the selection due to environment, which is everything else that's not species interactions. And the selection due to interactions also depends on the type of interaction. So in mutualism, we assume that interactions favor trait matching, so traits tend to converge. Whereas in antagonism, there's this asymmetry where the consumer favors trait matching, but the resource favors trait mismatch. So we incorporated this into our model of habitat destruction, and the first question we asked was, well, how does habitat destruction affect the coevolutionary trajectories? And so here I'm plotting. So here I'm plotting the variance of traits of each species in our meta network across the entire landscape. So each patch might have a slightly different trait value. So I'm plotting how variable this trait is across the landscape and how it changes with habitat destruction. So each line corresponds to a different species in the meta network. And what we found was that in mutualism, the trait variability tended to increase with habitat destructions, meaning that traits became more heterogeneous across the landscape, so more variability the more we destroy the landscape. But in antagonism, there's huge variability among species. So there are some species that increased trait variance and then reduced, but there are some species that continuously reduced their traits variability, spatial variability. And this was most likely because of, again, this release of consumer control, which meant that resources could evolve towards their kind of fixed environmental optimum values. And then the second question we asked was, well, how does coevolution affect the response of species to habitat destruction? And here we compared two sets of simulations. So we ran one simulation where we allowed the traits to co-evolve, so they were allowed to change. And then in the second simulation, we fixed the traits, so they were not co-evolving at all. And we calculated the difference in abundance of each interaction between the two simulations. And this is what I'm plotting here. So positive values means that the abundance in the scenario where traits were allowed to co-evolve was higher than when they were completely fixed. So here each point represents one interaction. And what we saw that in mutualism, co-evolving was always beneficial. So abundance when in the simulations with coevolution was always higher than without. And this suggests that the ability to co-evolve and to be able to adapt to changing species composition can actually dampen the negative effects of habitat loss a little bit. In antagonism, on the other hand, we also saw that the majority of interactions had positive, so an increase in abundance, but there were also some interactions that reduced in abundance. So this kind of suggests that in antagonism, maybe not all interactions will benefit from coevolution as in mutualism. So, I mean, this is of course a very simplified and general framework, but it showed that, I think it showed that overall antagonisms can be a little bit more unpredictable and how they respond to the changes in habitat destruction, and also how how their convolution trajectories can be changed by habitat distraction. So what I kind of tried to maybe show you today was how one type of modeling framework, so the meta-community framework, could be used to get some insights into a very practical problem like habitat destruction. And we went from single species to communities, from simple colonization extinction dynamics to including space, and then also including the eco-evolutionary dynamics. But of course, these models are still highly simplified. They have many limitations, and they don't represent the full complexity of real ecological systems. So what I'm wondering, kind of, whether these models can still be used to get some insights into these problems and really guide ecological design. So going back to the problem I encountered in engineering, where models can be used to guide engineering design. I'm wondering if maybe by incorporating a little bit more realism into these models, this could also be done for ecological design. So for example, taking restoration, can we design landscapes where communities are highly functional, they maximize biodiversity, but also maybe minimize parasite spillover? So I'm wondering whether we can incorporate more empirical evidence, like we heard today, into these models to kind of have slightly better or more realistic insights from these models. And I'm looking forward to your suggestions on this over this week. So thank you.
Does anybody have any question?
Thank you for the talk. It was a nice talk. When you model the space and the coevolution scenario, how do you impose the interaction? You put a limit in the space, or the species interact only in the patch?
They interact in each patch. So basically each patch can have a slightly different community composition due to the extinction and colonization dynamics. So at any given patch, at any given time, a patch might contain a subset of the meta-network. And then these species only co-evolve together in that little community, local community.
And they can migrate from one patch to...
And then they can migrate as well, yeah.
Okay. And they can be extinct?
Yes. So it's a stochastic process. So they have a probability of colonizing a neighboring patch, and also probability of going extinct at a given time step.
So when you increase the fragmentation, you can, for example, have some species that it's extinct for the whole landscape.
Yes, yeah, they can be. Because essentially, because they can only colonize closest neighbors. If you remove their closest neighbors, then they have nowhere to colonize, so then by chance they might become extinct.
Mm-hmm. Did you measure the extinctions in your landscape?
No, I always looked at the fraction of occupied patches, and of course as you destroy the landscape, that reduced that fraction. But the colonization and extinction probabilities were always fixed in the model throughout the process. So it was just about the opportunity of being able to colonize neighboring patches. But I'll also explain more about this tomorrow, maybe, if it wasn't very clear.
I think, I don't know if I got it correct. So you have like different patches, right? Mm-hmm. And what is the structure of these patches? How are they linked? Because you say the closest patches, but I don't know how they are measured.
Sure. Oh yeah. Sorry, I didn't explain that. So here, every pixel in this plot, so let's say this one, is a patch. And the way I model dispersal or colonizations is you can only go from your current patch to one next to you. So either this way, that way, or that way.
The four neighbors.
For neighborhoods.
For neighbors. Okay. And so when you're talking about that's why I got confused. When you're talking about the connectivity in this landscape, so you were talking about if there is a path between a patch that is on the top with the patch that is on the bottom part, is it?
Yes. Yeah.
Okay.
Exactly, yeah. So I don't know. Maybe here there's some white patches here, but they're completely surrounded by the straight patches, which means that they're disconnected from these white patches here.
But at each time step, you can only go from one patch to the neighbor. You cannot like jump from this one to the bottom one if you're not directly linked, is it?
Yeah, so that was the assumption in these models, but it's also possible to incorporate kind of a dispersal distance that, for example, reducing probability of colonization with distance, like an exponential kernel, something like that. It's very much possible to incorporate.
Okay, cool. Thank you so much.
Thank you.
Does anybody have?
Thank you, Clem. Great talk. Yeah, I have a question about these differences in abundance. For example, in antagonistic systems that you have. So each dot here represent a species.
In an interaction, but so here the units are of kind of abundance is the interaction. So I was interested more in interaction than species, but the trends for species are very similar as well. So basically, I classify. So each interaction in a network is accounted. I see it like the same way I see different species. So this particular network has, I don't know, that many interactions, as many points as there are under the box plots. Don't know if that makes sense.
No, because I was trying to understand the dynamics in antagonistic systems, and well, yes, because you have positive effects in abundance for the consumers, for example, but maybe for the resources they're gonna have higher abundance when the trades are fixed. So so below this line you have the resources, and above the line you have the consumers, or no?
Well, so you're right that for example the resources will benefit from the absence of the consumers, and they'll probably benefit more from the trades being fixed, but no, each point is an interaction, so it's still it's an interaction with two specific species to so basically I'm counting in how many patches interaction between species X or consumer X and resource Y was present.
It's like the occupancy in the landscape.
Yes, yeah.
Okay, thank you. So I'm still like super curious about the trait variance thing that you have, like this too. Yes. This is super kind of interesting to think about, because in antagonistic systems looks like things are much more unpredictable than in mutualistic systems, but did you try to figure out some relationship between these spatial variants in like trait variants and where each species is, like the structure of the spatial network, or there's no relation?
I didn't see any very strong trends between. So I simulated this with a number of networks. I think it was 20 different networks, and they all had different structures. But I didn't see any strong trends in this. So the results looked similar for all networks. So yeah, didn't.
Okay, thank you, Clem. Thank you.
thank you everybody that was here today with us, both physically and all that was with us online as well. And now we finish our symposium for today, and I just want to give a notice for those who will be with us during the week. Can we say in which room we are gathering? Or? Yeah. I don't know how to turn it. Ah, okay. It's already finished. Okay, thank you. Bye bye. Thank you.